এটা
দেখ খুলে দেখ। দাদা
এইবারে হয়েছে, ঝামেলাটা ফিক্স হয়েছে।
হ্যাঁ, চেড়ে তুলে পেছনেটা দিয়ে দিই নাকি?
এখানে দিদি
এটা তুলে পেছনে
হবে না,
একটু ঘোরাতে হবে। এইটা দিয়ে ঘোরাতে নাকি? এটা কি এটা দিয়ে তো ঘোরাই। হ্যাঁ এবার বলছি এইটা একটু পিছিয়ে দিই।
আরে হ্যাঁ হ্যাঁ দেখা যাচ্ছে
সর আচ্ছা,
ও চালাই হচ্ছে না।
এখানে রাখি,
কোথায়?
এখানে রাখি।
কেন?
দুটো আসছে না।
ও তাহলে একটু পেছো, তাহলে একটু পেছো।
যা তো পেছনটা যা রোহিত।
আরে পেছনে
চেয়ারটা সরাতে হবে। হয়েছে, সেট কর, যা সেট কর পিছনে।
চেয়ারটাকে পেছিয়ে দে আর আসছে
পুরোটা ওইদিকে অতটা গ্যাপ লাগবে না।
দিকে সর, সোজা কাছরা কাটা সরা।
আবে গান্ডু এটা দিয়ে সরা না।
না না সরাইনি, এটাকে ঠিক করছে।
আচ্ছা লক কর। আর এটাকে একটু এইটার সাইডে এত ফাঁকা লাগবে না বুঝলি, যাতে দুটোই দেখা যায়। হ্যাঁ পারফেক্ট। আরে এইটার সঙ্গে তো গ্যাপ গ্যাপ
এইটাকে যদি একটু ঠিক করে হেলা আছে নাকি এখানে বস কি হেলা?
না ঠিকই আছে হচ্ছে।
ঠিকই নেই রে, এখানটা এই যে তোর এটা আছে ওই জন্য এটা হেলা দেখাচ্ছে, এইটা এরকম।
তুই ওইখানটায় বস।
কোনটায় বস?
তুই ওইখানটা, আমি তাহলে ওইখানটায় বসি।
না
না ওই জন্যই
এটাকে লাগবে। ও বুঝেছি।
আরেকটু বাস আমি নিজেই বাচ্চা।
I would request all the students to get seated. We'll be starting with our session now. We'll be resuming with the guest lecturers. To start this session, I would request Professor Deepak Kumar Jana sir and Professor Shomitra Kaur sir to take their seats as the chair of this session.
মানে মারপিট করে দাঁড়িয়ে কার ট্রাইপডটা কাটছে না।
ট্রাইপড নিয়ে অনেক ইয়ে হয়।
বাঁকা আছে।
গুলি চালানো হয়।
ধীরে হেলায়।
The next speaker for this session is Professor Devashish De, Professor, Department of Computer Science and
এবার
Engineering,
ভালো হচ্ছে।
Macau. So would you please kindly come up on stage? Devashish De sir is a professor of the Department of Computer Science and Engineering at the Maulana Abul Kalam Azad University of Technology, West Bengal, India. He received MTech from the University of Calcutta in 2002 and a PhD from Jadavpur University in 2005. He is a senior member IEEE, fellow IETE, and life member CSI. He was awarded the prestigious Boycast Fellowship by the Department of Science and Technology, Government of India, to work at the Heriot-Watt University, Scotland, UK. He received the Endeavour Fellowship Award from 2008 to 2009 by DEST Australia to work at the University of Western Australia. He received the Young Scientist Award both in 2005 at New Delhi and in 2011 at Istanbul, Turkey, from the International Union of Radio Science, Belgium. In 2016, he received the J.C. Bose Research Award from IETE, New Delhi. In 2019, he received Shiksha Ratna Award from the Government of West Bengal. He established the Center of Mobile Cloud Computing, CMCC, for IoT applications. He is Vice Chair of Due Computing STC of the IEEE Computer Society. He published in 360 journals and 200 conference papers, 15 books, and filed 10 patents. His H index is 38 and the citation is 7,000. Listed in the top 2% scientists of the world, Stanford University, USA. His research interests are in nanobiotechnology, IoT, and quantum computing. He is the chairman of IETE and CSI Kolkata chapter. Today, Professor De will be enlightening us about the topic Internet of Bio Things. So, please kindly take the podium.
Thank you, madam.
Before you start with the speech, I would request Srishti Lata from second year Department of Biotechnology to felicitate sir.
Good afternoon. Are you sleeping? Good afternoon. Thank you. So good to see a lot of students are there. And today, next 30 minutes, I'll be discussing on the topic of IoT. And where IoT, anyone know what is IoT? You heard? Internet of Things. Yes. What is the purpose of IoT? Why IoT? IoT is important for, this type of thing. Can you give any example of IoT? Anyone? Any example? Everyone uses this IoT almost every day. Yeah, smartwatches through which you can control everything. You know that there are various apps are there. In your house, you can make your switch AC on, right? You can switch off your light at home. If you have a smart home, even if you go to the market, Philips or many companies, they are preparing these circuits, switches, which you can control it through your mobile phone application. You know smart TV, right? You can control your TV through your mobile phone itself, right? Nowadays, we can switch on and switch off our AC. Lots of ACs are there, right? And anyone can control from long distance. Okay? And it's very much important. Why? Because we have to have a full control over the system. Okay? I can give you one example that my brother stays in US, New Jersey. Now when he comes to India, he says, I've seen that he's always talking to Alexa, switch on the heater, switch off the heater. I said, why is switching on and off? Can anyone say anything? He's saying that switch off the AC or heater or all these things. Why? Why they are doing it? When he is in Calcutta, he is controlling his room's heater. Because, when there is no heater, then what happens? Then water pipe will be frozen, ice. Then what will happen? The pipe will burst. Agree?
Yes.
Then when you return home, after 1 month, you will see water is locked. Water locked home. Everyone will be under, you understand what I mean to say. So, IoT is not a fancy thing, it's a need of the hour. Agreed? Yes. So, we have to control the whole system through our IoT. Clear? So, he is monitoring the temperature of his room so that the pipes are not blocked. So, in our day-to-day life, we are using this IoT. Actually, we are controlling the things, Internet of Things. Here also, right? Things means anything which is connected to Internet. For example, you use Ola and Uber app. Through this is also IoT. You are controlling the car to your address, and you are giving a destination address. See, you are controlling the car, and this car is the things. You are controlling the mobile phone itself. So, mobile phone data is going to the cloud, and that cloud data is going to the car, and you are controlling the whole system. Clear? Yeah, this everything is there. You have seen that movie of Gupika in Bhagavan? Yes. You know that through the claps they used to get the food, right? Monda, mithai, everything is coming, right? So similarly, you know that through the Zomato and other things, you are getting the food delivery. So everything is available through IoT. We are getting all the connectivities and control over the whole system through IoT, right? Now the question is, today I will be discussing on the things called Internet of Biothings. Biothings, I will just concentrate on the biological products or biological things. Biology means here, you are from Biotechnology, right? So we will be considering the biological systems, means human being, animals, how to control the human being, everything. Okay? So extra term is added, IoBT. Instead of IoT, we are calling it IoBT. And you will see, this is a lot of lots of applications are coming up for IoT applications. Okay, one of the things I can tell you that one of my students, his name is Siddharth, is from our lab, diploma, and he is working in Taiwan, and he is developing a device for sleep monitoring. When you sleep, whether you sleep properly, whether the sleeping is sound or not, that device will tell you. You have to wear a device on the head, lightweight device, and through a smartwatch you can see how much, whether you are really sleeping or not. Are you getting a point? So if your sleep is not proper, some pulse or some sound or some light music will play, so that you will go to deep sleep. So not only it will find out whether you are sleeping or not, if there is any sleeping disorder, it will also correct it. So a lot of research is on, because we are now in so much tension, right? Every day we have a lot of tension in our life, and we are not sleeping properly. So sleeping is very important, agree? Sometimes it is written that 8 hours sleeping is okay, 6 hours, a lot of debate is there. But the question is, sound sleep is very important. And that can be also controlled. Amazing, isn't it? And this applications on human being, and they are developing the devices. That is the brain-computer interface, and we can control it, these BCI devices, through internet. That means the person, Taiwan lab, I think Lyoko, one collaborator, they are wiring the device and connected to your doctor, and doctor can monitor how many of the patients are really sleeping. Any problem, they will call you, and accordingly the medicine or the system will start, the process will start. And these are very interesting applications of biotechnology, where the various health parameters can be discovered through the brain-computer interface. Means you are wiring a device, and through that device is data UV cloud, all the cards are connected, you are calling the UV, right? Similarly, doctors can also see how many cards are there, what is the delay, and what is the cost, other factors. Similarly, in this case also, doctors can see how much amount the patients are really sleeping or not. Okay, that is very important for your health issues, right? You know, there's not only the sleeping, other health parameters can be also detected, right? Especially using the smartwatch, Apple smartwatch, you know, it can measure everything. If you are not drinking water for say less than 2 hours, you know, so what will happen on the skin? Anyone can say? You're from Biotechnology. If you are not drinking water for 3-4 hours, what will happen? On your skin effect, any effect will happen? Yes? Drying or itching kind of thing. So the smartwatch can sense that you are not drinking water. So they will ask you, instead of time, it will show you drink water. It will recommend you to drink water. Sometimes we are so busy in the meeting or in a classroom or in lecture or some other reasons, we forget to drink water. So these things will recommend you so that your lifestyle increases. What is important in our life? That our life quality will be improved. Agree? And IoT actually helps to improve the quality of life. I am repeating, IoT helps to improve the quality of our life, and that is very important. Okay? Now, what is the agenda today? I will be discussing on some biosystem engineering part, Internet of Health Things, biosensing, and edible electronics or edible IoT, and then we will be discussing on agricultural things, some of the plants and other things. So IoT has lots of areas, especially in the biology area, and agriculture, healthcare, robotics. You know, most of the surgery is happening through robotic surgery. Okay. And some of the things are happening, you know, you heard about haptic computing? Haptic music, you heard? Haptic. How this robotic surgery is happening? Anyone can show? Doctors will be wearing a gloves. Yeah, that is one thing. Another thing is that, so doctors will be wearing some gloves, remote control things, and they put the surgery things in, and they are moving their hands on the air. Everything is called haptic. Haptic, H-A-P-T-I-C, haptic computing. Okay. Haptic computing is very important for surgery. You know, distance surgery, doctors are using this IoT applications. You heard about distance surgery, very popular, and it already started in India also. Okay? You know that when you used to hire the musicians, okay? Say, who is the best musician in India? Or director, A.R. Rahman? Say, suppose A.R. Rahman cannot come here physically, but you can play some guitar. From remotely, A.R. Rahman play the guitar here. Possible. And it is possible through the haptic devices. Means the device we have developed, Tomatit, and we are all working on it. The device will be there, guitar will be there, with some sensors, IoT controlled. A.R. Rahman from anywhere in India or anywhere in the world can play the music. Okay? So in many things can happen through the safety computing. So IoT is bread and butter for modern day industry, and everyone uses it every day. Okay. So IoT means here Internet of Bio Things, where actually we are connecting the biological world to the cyber world. Okay? Our biological world to the cyber world. Cyber world means the cloud. Everyone knows cloud. You use Gmail, you use Amazon cloud, Google cloud. So we are actually transferring our data from our health data in the cloud, right? When you are wearing a smartwatch, if you are connected to certain hospital, say Apollo Hospital or Amri Hospital or say Medica or any other hospital, then your data will be registered to certain hospital. If you have any abnormality, then the healthcare givers, they will call you and they will say that, okay, you have some abnormality, you can come for checkup. If the abnormality rate is high, then what will happen? They will send an ambulance. You know, there is a very important word in our life called prevention is better than cure. So you have to have the ambulance first, right? And IoT gives this sentence clearly that you must take care of your health properly, right? So you understand. So IoT is already part of our healthcare system. You know that you call it family physician, right? So you go to some doctors because their doctor know you very well, close to your area. So maybe some this kind of hospitals will be there. Everyone have to register. It will be mandatory. Okay? In many countries, it started mandatory. You have to register yourself to certain hospitals so that they will take care of you 24/7 basis. Not only during the time of your visit. Okay? Through the smartwatch, they can get every parameter. Your blood sugar, BP, everything can be measured nowadays. Isn't it? All the parameters are possible to get. So we are connecting to the biological world to the cyber world. There is a very interesting paper called Body Cloud. Means body parameters are actually going to the cloud, and cloud is taking decision to the IoT that what will be the next step. Course of action. So preventive healthcare, very important term is preventive healthcare, right? So it is only possible through this Internet of Biology. So you know this already I mentioned that we are wearing some sensors. We call it wearable sensors, right? And where we are actually using these wearable sensors so that our data will be going to the cloud easily, right? Smartwatch, now we think smartwatch is only for fancy. Just wait 2-3 years, everyone have to wear the smartwatch. And cost will go down below 1,000 rupees. I don't know what is the present cost, maybe 2,000 or more, 3,000, 5,000, whatever. It will be, just wait, after 2-3 years, it will go down below 1,000, and everyone have to wear the smartwatch, and everyone will be connected to certain hospitals or health centers, right? So that every people can get 24/7 analysis. So if you see this picture, so there are within the body sensors, wearable sensors, this is the hospital access point. So we are getting the sensors actually, various sensors, hundreds of sensors. So if you have some particular problem, some people has a problem in the leg, bone joint problem, pain problem, back pain, neck pain, spondylitis. Special kind of sensor they can put. They do the posture correction. Posture correction. Godrej company, Godrej company. They develop certain chairs. If you do not sit in the chair properly, some big sound will come, some alert will come. And that is possible through IoT. How much time on the day you sit properly on the chair? So posture is very important. What is the height of your monitor? How you are looking at it? So posture correction apps are coming up. So these are very interesting applications for healthcare management. Also, those who are sportsmen, sports person, they are also that skill development is very important. So you want to play like Maradona. Maradona is no more in this world, right? Messi is here, right?
Yes, sir.
Ronaldo is here. So to follow them, then what will happen? You want to build your skill, then you will take a football from Messi. Skill is developed by the football. You will be using it, and the football will, when you will be doing a skill practice, the football will tell you there is a screen, or from the football there's some on your smartwatch to show how much amount of skill you have developed. Whether it is 60%, that means you have developed skill of 60% of Messi, or 70%. So you keep on doing it, you'll see the skill will be accurately up to that level. You can reach to that level. Have you seen this World Cup, last World Cup? This football World Cup, right? So every football has an IoT device. Whether the Ronaldo's, that controversy, that hit, there is no touch. It was detected by the IoT device. So there is a lot of sensors are there, right? Now these footballs, when you go to the market, you will buy a football, not only normal football, Messi football, Ronaldo football. You are getting a point. The skill level is set up there. Or you can buy a football, IoT football, you can set the level to that level. Based on the software, based on the skills. So, skill can be also developed. So, it is only possible through these biosensors. And especially, this kind of sensors are very important for senior citizens. Or, we are calling a very interesting term called royal aging. Royal aging. See, earlier our death around 68 to 70, right? Nowadays, it has increased 80, 85, around 80. At least above 75 or around above 70, right? It is only possible because of preventive healthcare. So we are calling it royal aging is only possible. So everything can be monitored, especially the senior citizens stay at home. We are always at office or some other places. So they are being tracked by multiple sensors, healthcare sensors, to find out the solution. Now, the question is, what are the components? First component is biothings. Means I am the thing. When we are talking about healthcare, I am a thing. Means I am the object. I am the thing, right? Agree? And we are calling it biothings. Second is interface. The smartwatch is the interface, or the smart sensors are the interface. Second step. Third step is the gateway. So you have access point. So in this room there is a internet. Yes or no? You get internet access point in this college? I don't know.
Yes, sir.
So if there is an access point, we can transfer the data to this device. And this medical service server that is in the hospital. So these are the 4 steps. First is the human things or body things. Second is the interface. Third is the gateway. Fourth is the server of the hospital server. So these are the basic 4 steps we have to follow for data transfer. So there are a lot of applications in there. So for IoT we need continuous internet and continuous cloud access to various applications. It is totally app-based, like your Uber app, Ola app. So total healthcare is now controlled by the apps. Even various critical diseases can be detected. So one of them is you've done endoscopy, right? So endoscopy you are eyepiece inside, or sometimes you are taking a tablet with a capsule with a camera, and they are taking the internal image. Doctors or the nurse, they can monitor what is actually happening, and this capsule will dissolve within say 6 hours. You have to stay in the hospital, and that capsule can be also controlled. I was talking that, It is programmed in that way. So lots of healthcare research is going on. It's a very interesting area of biotechnology. I am from a computer science background, but we need the help of your side so that it can be integrated. It's an interdisciplinary problem. So we have to work together. Right. So there are a lot of challenges there, that transmitter receiver design. India, we are almost in zero level. All mobile phone, no mobile phone is developed here. Electronics, we are in zero. But largest consumer in India, but zero manufacturing, unfortunately. Manufacturing is done in Taiwan and other countries, right? Mostly in Taiwan, Singapore, those places. So the cost is a little bit big factor. Transmitting area, power, safety. Safety is very important. Radiation, you should not give more radiation. And so I'm just showing you one application, what we have developed 2008. So what do you need? You know that you heard of NICU, neonatal premature baby, where they are in the hospital, they put in certain chamber in certain temperature so that the baby can grow properly. Agree? So if you see 2008-9 newspaper, some baby were burned in the NICU, Narjikar hospital. It was in the newspaper. So that time I thought what is happening there. So the baby comes with a wire. I was talking to them. Baby comes with a wire, and baby, just premature baby, how they can connect them to the wire. And when they're throwing the hands frequently, sometimes possibility of the wire will go outside hand or outside leg. So what we did, we converted that total NICU to wireless NICU. Means we are monitoring the temperature through wireless band. Just a wireless band, there is no cable connection. Totally wireless. Okay? Either through image processing or through wireless connectivity. And this is totally, the NICU is converted to IoT enabled. Means we are measuring this temperature. There are certain parameters, well-known parameters there. Say around 25, 36. So what is the year and age? Accordingly, we have to monitor. So we develop the apps based on the weight of the baby. We can control the temperature, and you can see the temperature, heart rate, and acceleration. If the three parameters is okay, then health of the baby is okay. So doctors can monitor it from their own home. They do not have to stay in the hospital. That is the beauty of the whole system. And we developed, I don't know that time Ola Uber was not here. So it was very tough time, but we did this experiment around 10 years. Also, we did some experiment with C. elegans, very interesting animal, I think you know. C. elegans, we have C. elegans in our university. With biotechnology department, we did this experiment. C. elegans is a small worm, right? And why C. elegans is so popular? Yes? It is? Yes.
It leaves a silver line where it goes and
No, why we do more experiments with the C. elegans? C. elegans is a worm, and we are a human being. For drug delivery, studies expert will give the drug to me. I think you are expert in drug delivery, drug design, all these things. The C. elegans. No, transparent is one of the big feature, but important thing is the gene sequence this almost matches 80% homologous with the 80% matches. Almost 80% matches. And this C. elegans research got 4 times Nobel Prize in biotechnology, in biology in India. 4 times Nobel Prize. You see the things in Wikipedia or internet. So I was fortunate to do this experiment with this C. elegans. And what we did actually, we have seen the lifestyle and we have monitored and controlled the growth parameter, everything through IoT. So this experiment is done by Biotechnology department, and what I did, I controlled the data, imaging and Joy Bandhavandha madam is there, and we have controlled the data, and one of our research colleagues was there. So he is taking the data, and we are controlling it we can see the movements of the C ligands on the assay very easily, and the control of the C ligand's path, movement path, that can be also very easily tracked. And it is only possible using this IoT. So through this IoT, we are actually measuring the movement, number of C ligands. There is a computer vision applications there, through which we can measure this. So C ligands research, those who are interested in drug delivery and drug processing activities. So we did it with carbon nanotube, and we also used this carbon nanotube DNA molecules so that the toxicity is reduced. I'm not going in details, but this is a very interesting area where C ligands can be controlled using this IoT. The movement on a plate, around hundreds of C ligands are there. We can control the movement using the laser light or by the internet. Both are possible. So this is the movement, the tracking pattern. Okay, this is another very interesting area where Amit is working. That Amit is here, your teacher. Now he is working on level 3 biosensing. Means, see, nowadays for a sugar test, you have to go to any diagnostic center, yes or no? Everything kit is available, isn't it? Sugar test. Also, BP monitoring, all this health monitoring, nothing is required. Everything we can do at home. Even Apple smartwatch gives much more features. Samsung also have started a smartwatch that also gives lots of parameters without taking any blood sample. Very interesting. And these are all possible to measure the systems. So, what is biosensor? Actually, biosensors are, it's called level 3 biosensor, right? You can measure the molecules presence. I think you do a lot of sensing in your lab, measurement. So, you have to develop certain structures which can feel the presence and tell what kind of molecule it is, right? Whether it's a DNA molecule, whether it's any other kinds of things. So everything can be done here. What is here? Say suppose this is a molecule and this is a receptor, this is a transducer. So it will give some signal. Signal will tell you what kind of molecule it is. Transducer means which converts non-electrical signal to electrical signal. Basically, this is part of sensing. Sensor is like that. So this is a part of detection of the molecule. So see, these are the things I think everyone knows. So if you see, this is a very interesting that samples, these are the samples, and these are receptors, okay, bioreceptors. So there are enzyme, antibody, these are all your biological sample things. And this part, you are expert in this domain. And signal processing part is through electrodes or pH sensing. And third part is signal conditioning. And last is electrical signal. So this means this will detect what kind of molecules are coming. So there are 4 stages. Sample. The second is bioreceptors. Third is signal transducer. Then signal conditioning. What you are getting the signal, what kind of molecule it is. What kind of system you are using, right? So these are the 4 steps for designing of the biosensor. And there are various structures there where Amit has done that various kinds of biosensing. This is a silicon-based system, and this is a Pershing gaps. The gap size is equivalent to your molecule size. It can be several nanometer, right? How much? 10, 15, 20? It varies. Every molecule has different size. Anyone is interested, you can talk to Amit. He will tell you. So these are the various interesting sensing applications. So there are various structures. I'm not going in detail. We can calculate the sensitivity and selectivity of the product. These are the semiconductor products, right? So yes, we have did a lot of transactions, actually transactions based on these devices. Okay. Now see, we are talking about this conference name is what is the name? New Horizons of Biotechnology. See, have you heard about this thing?
Mushroom.
One of my friend, I think Biplav is there. He is also in computer science. He works in the domain of actin automata. So this is the work by Andrew Adamski. This month, March 2023, he has developed the CPU, means Central Processing Unit, using mushrooms. I'm not going into detail. Anyone is interested? We work with. He's my friend, very good friend. He's senior to me, but he always said using the mushroom network, you develop the CPU, Central Processing Unit. It is one of the highest cited paper in the Nature. No, highest downloaded, not cited, because just published. Mushroom-based computing, right? The new areas of biotechnology. I think Biplav also works in actin automata. Actin means actin molecule, actin myosin. So this is known as collision-based computing, right, Biplav? So where he did his PhD on Biplav did his PhD on actin automata. Automata means present state to next state. Present means present time to next time. So it is lots of molecules are collisions are happening, and based on the collision, different logics are developed within the body. Suppose there is a mosquito in my left hand here, right? So mosquito bites here, automatically brain will give a signal to. So this reflects, and the logic comes through actin myosin. We also studied the computational behavior of this actin automata using this actin molecules and myosin molecules, right? So Biplav did his full PhD on this biological computing, means using actin. We call it actin automata. Right? Also, you can see what is this? Shoe shows, right? Lot of fungal things are happening, right? Agree or disagree? Lot of bad signals come, right? If you do not agree? This paper is published just yesterday. I thought I should put it. This is also by same group, Andrew Adamski. He is in Bristol, I think. Where is Bristol? Not Lower England part, right? So he did a network using a shoe sole to find out the pressure point. Pressure point. How, based on that, your measurement of the health parameters can be done from your shoe sole. Right? Very interesting area of research. Very interesting. This is a combination of computer science and biotechnology. So this is the Biplav's work where he did it on actin computing for various. We can connect it to IoT also for various actin applications. We did one board for stride analysis. You know stride analysis? Anyone know what is stride? Stride, walking pattern, right? Yes. So I met a doctor. One of my students was there. I mean, his father is a neurologist. Okay, very interesting story. And I was there for a coffee, and he said that he asked me to walk, just to walk for 6 feet and rotate and come here. 6 feet only, 6 to 10 feet maximum. And by the walking pattern, he said, what kind of disease you have? This is he is one of the experts in Calcutta Medical College. He was the head of the department of neuro systems, and using the walking pattern, we can measure what kind of health disease you have. So what we did? Now, we use their domain knowledge. One of my students, Amarto, and Pratiti, his father is doctor, Pratiti Bharti. And what we did, I'm just showing you, we have measured, a lot of people are working, how they're working, the working pattern can be transferred to IoT. Means, what I did, you know that person is working, we use a drone, flying drones. We're moving around the person 2-3 times. Just by looking at the working pattern, they will tell you what kind of disease you have. So, if you have any disease, If a patient at your home, so drone can move from any hospital. In-house, it can detect what kind of disease you have. Are you getting my point? Agree or disagree? So these things can be resolved. So we did this experiment and we got very interesting result. We have validated with our results that using this various points on your leg, these shoes, the walking pattern, through that we did this stride analysis. Using the drones, we did this real-time stride analysis. Real-time means how you are actually moving. Through that, we have detected the disease. Okay. Another thing I must mention you, how you buy the shoes, how buy your shoes. What is the size? He says 9. Do you agree? I say no. Your approximate size is 9.
Yeah.
Madam, you will agree that both the size of moving is not perfect. Agree or disagree? Huh? So I can tell you my one experience. I worked long time through computers. I have a lot of shoulder pain. I went to a doctor. I'm not telling the name, but he said he just put some pressure point on the leg, and he says, okay, my pain disappeared. Because our pressure point is very important. So we are wearing, listen, in India, we have to go to Bata shop or Hadiths or anywhere, we are buying the shoes. Right? It's a wrong thing. If you go to China or many other developed countries, where you have to go to the shoe shop, you have to give your both the legs, and you see, madam, at least 60-70%, maybe more than that, your leg, both the legs are not same. Maybe someone is 9.1, one leg is 8.7. Both the legs are not same. Everyone become very conscious. So what is the solution? You have to go to the shoe shop. What is actually happening? You go to YouTube, you will get it. You go to the shoe shop, give you both the legs, and your measurement is done. Second, you go to a smart screen to choose your color, to choose your straps and other color sizes, and just pay the bill to your cards or whatever. Then what will happen? After 20 minutes, your shoe will be ready and delivered to you. Okay? So, what I mean to say, this shoe is very important for our life. If you are wearing a wrong size, that will affect our health. In the reverse way, what we are doing it now, we are, again I should not say, right? From not sir? I should call it. Our current research is on the shoe-based IoT research. I'm not going in detail. But why I am telling you, then health parameters will come, better health parameters. When you have to change the shoes. I have seen some Bata advertisement that someone is wearing a shoe for 25 years. Is it correct or wrong? Wrong, because the shape will change. Sometimes I have seen people are buying some 20 rupees sole and putting it inside. That is a totally wrong idea. So I have to tell you, so you have full protection. Your smartwatch will be connected to the shoe and you tell you what kind of health disease you have. And most of the health diseases can be resolved on the shoe part. One of the Chinese group we are working, I'm not going into details against IP problems today. So shoe is important for health analysis, right? Last part, I just like to mention, something is mentioned by Comrade Moni that we are doing this online, agricultural things using on the rooftop, right? Using this IoT. So what we did a solar panel, we did some IoT device which can measure temperature, soil moisture, turbidity and pH. These modules we have developed. I'm just going to the system. Electronic circuits which can measure the full system. controlling of the sensors, this management means alarm, means any kind of message problem, then alarm will come. So these are the full system. Sensors, gateway, internet, and the control system. So these are the four stages of agricultural control. Also, we need something experiments on fruit ripening. Apple. I was in Kashmir, and Kashmir is famous for apple. Have you ate any Kashmiri apple?
Yes, sir.
Hardly you ate. So beautiful, I never got this kind of apple in Calcutta. It was like a mango. I was eating apple here in and you cannot believe it. How much taste it is. In Calcutta, we call it gala apple, very costly. 300 rupees or 400 gala apple. It smells like it. So beautiful apple. But there is a problem of that apple. If you pluck it before time, it's not so much tasty. You put after time, you cannot eat it. Not it. So there is a best time. There must be a best time to pick the apple from the garden. We did some experiment. We have one project of Techie project, I think World Bank project. So we are connected to the IUST, Islamic University of Science and Technology, Kashmir. There is a food technology department is there, like your biotechnology department is there. This project is with them. So we did a circuit. I'm not going in detail. It's a very simple circuit. Apple is a simple circuit with 5 volt voltage. These are the samples we took, and we measured the value with various cases. And see, this IoT samples, the Raspberry Pi controller, to the cloud we are getting the data. On the mobile phone, the farmers will tell you what is the best time to pick up the apple. We developed it, successfully did it. We have to patent for it. Okay. So, apple ripening index monitor. And this is used in Kashmir. Also, Kashmir, another very interesting area is what is called red color?
Strawberry.
Saffron. I was in Pulama. Pulama is famous for different reasons. Pulama is famous because of saffron. Pulama is so much in the news because of saffron. Only saffron in India is developed in Pulama only. And so much small trees. We did some, but those trees are getting infected by the various kinds of space. Yeah, that is the problem. We did some experiment out there also to give the pesticides on the thing. So this is apple industry and saffron industry. This is very much popular in Kashmir as well as in India, right? Very costly. Saffron, very costly. It's a very beautiful pink color, yeah. Yes. I cannot tell you. You go to September, you will never come back to Calcutta. So beautiful plantation there. So yeah, these are the measurements we did. Real data. It's our data. I'm not going it. So this one of the publication is approximation of ripening for using IoT devices for e-healthcare. So you must eat healthy, right? So not only for picking up, when the apple come to your house, you can also see, you can pick the samples, means there is a device like this, you put it, best time to eat. Today or tomorrow or now, that will give you the recommendation. We have the device. This is another work we did in Shillong. There is a conference going on today. I was supposed to be there today. Okay. So what we did? Very interesting work we did there. Farmers in the hilly area, hill steps are there. Hill steps, right? Farmers in the hill steps. What is actually happening there? If there is excessive water or step permits are there. The problem is there are no control of the system and very difficult to go to those areas. So we did long distance Wi-Fi link network. This is one of the highest cited paper in actually transaction of IoT, more than 400 cited it has. So if you see the paper work, right, we are simply gathering data from the farmers, okay, and there is a link See, 10 kilometer. Long distance data you can send. So this is the first required with short range, means within this room we can send the data to the cloud. Long range. So these are the apparatus. This is the new, not this Hill University, Shillong, to the river, means the hill beds. These are the experiments, these are the devices through which we are sending the data. This is the IoT device, which is on the plantation part used by the farmers. And we did this experiment on the successfully long distance Wi-Fi. It's a main project in Ministry of Electronics and Information. We also developed, I'll skip it within 2 minutes, I'll finish. We also developed this Box, a quad sensor, means we are using multiple sensors. We can combine multiple sensors together, we call it quad sensor. Advantage is, if you have 4 decision makers, what will happen to the problem? In a say you are advertising a conference. If you have a 4 decision maker, what will happen? Madam? 4 different opinions. Okay, he will be the internet speaker. No, he will not. A lot of idea will happen. Decision maker should be 1. So you have 4 sensors, 4 kind of results will come. So it is confusing. Agreed? So must be single data. 4 sensors, but data must be single. So must be some aggregation. So we did this aggregation here, and 4 sensors together, and farmers can get the data. This is the circuit. This is the actual system, and we published in IEEE Consumer Electronics magazine, and this is also patented. Commercial. We got the patent. You said agriculture things cannot be patented. Yeah. Yeah. Okay. So we did a patent audit 2 years before. Also for hydroponics, within the house we can do this hydroponics. Everyone know, right? Aeroponics, hydroponics are very popular. So no need soil-less technology, and totally IoT control. See, these farms can be controlled by IoT. This is a picture in my house. This is in my house. This is a real tomato, potato. And these are temperature, pH, all these things you can do this. In the kitchen, we can grow the fruits using IoT. And moreover, recently we are working on how to use this artificial intelligence for these bio devices to grow and control. So conclusion is that IoT is important, and we need to develop together. See, my knowledge on biotech is limited, right? So we need to collaborate, we need to develop the products together. Otherwise what will happen? You will not get a good result out of it. Unfortunately, we do not collaborate, we want to do it alone. Nothing is yours. We came to the world only for certain years, that's it. We have to do for our country only, right? So, sustainable consumer electronics products is must be. And generally, we can feel proud we have done something for our country. Okay? Thank you very much.
Wow.
This is the time for question and answering. So,
Sound effect.
So, are there any IoT devices through which we can measure some specific quantity of nutrients in a particular place in the soil for ease of farming?
Yes, we did it. I was working with Professor Ram Gopal Rao. He was professor of IIT Powai. I just came yesterday from Powai. Okay, we did the experiment in Powai to measure the pesticides and agricultural elements from the soil, and we are printing those sensors on the postcard, Indian postcard, 25 paisa, right? And we are sending the postcard by writing the address to certain farmers, and they are putting the postcard on. They can say what kind of animal problems are there in the soil, whether it is excess fertilizer or less. So in half an hour or 45 minutes, I cannot finish everything. But this is a very interesting question you have asked. It is MEMS, based on NEMS. NEMS means Nano Electronic Mechanical System. NEMS. Yes, very interesting. That is happening. And Professor Ram Gopal Rao was a of IIT Delhi also. So they developed, I was also part of the team, but he is the main person to develop this. It is already there. And you'll be happy to know, after using this, in Punjab, they use X fertilizer. And in Calcutta, West Bengal, it is opposite. So after using this, soil health is very important. You heard about Sadhguru?
Yes, sir.
He has a movement. I met him last December. We have discussed on water and soil. I asked for appointment. He came to Calcutta. So he gave me appointment only for IoT device for water and soil. So I cannot discuss here, but if you are interested, I can tell you the details. So he did a lot of movement, and this kind of sensors are very, very popular. Only 25%. And government is funding. Government of India is funding.
Thank you, sir. Sir, I have one question. Sir, can you tell me, like, can IoT be used for detecting genetic disorders in the fetus which are present in the mother's womb, or for detecting certain kind of neurodegenerative disorders, say Parkinson's or Alzheimer's?
Yes, it is there already. Various health parameters and monitoring systems IoT is using, and these things can be detected. Even I can tell you another thing that one of my friends, Nidhan Mishra, Nidhan, is working. He is one of the famous for COVID detection. He is from NIBMG. NIBMG. You have seen in television many times. Even he can tell you through your genome sequence analysis, using also IoT and other things, that after 30 years what will happen to you. Future can be predicted. Because everything is written on the gene. I think madam can tell you.
Yeah, Ultron.
Everything is written on the gene. You can predictive lifestyle modification that can be done. And the IoT is coming here, human genomic sequencing. It was earlier 1 crore rupees per human being. Now it is 20 lakhs. Now it is 1 lakh. Our target, I am also working in the project. Our target is within 10,000 we will do it. And Government of India, if you please visit NIBMG in Kollam. Madam, I think you can arrange a visit. I can arrange. They will get to know what are the very interesting things that happen. My friends are there. And there is specific one-day program for Saturday for students only. If you go there, you will learn a lot. So just like your X-ray, we need blood sample, sugar test, and if you do the genomic sequence of your body, okay, you can also predict how much your life is. That can be also predicted. Disease can be predicted. Accordingly, you can do the precautions.
As we are running late, so thank you, Professor Devashish Dey, for delivering very nice lecture. And once again, thank you for accepting our invitation from India to Macau. So we are very regards to you for coming in India Institute of Technology. So thank you. Now we over to Dr.
Thank you, sir. I will now request Dr. Tilak Raj Maiti, sir, to please honor our esteemed speaker with a token of appreciation.
Oh yeah. চারটে লাইক পড়েছে ভাই ফিলিং প্রাউড
সাবস্ক্রাইবার 10 খানা সাবস্ক্রাইবার বেড়েছে
অনেকেই থাক তাহলে বল
মনিটাইজেশন অন কর
আরে পড়বে দাঁড়া না তুই
We will move on to our final guest speaker for the day. He's someone who is already familiar with the premises of Haldia Institute of Technology. A very old alumni. It is my pleasure to call upon stage Mr. Abhijit Ghorai, sir. A few words have been composed in his honor, stating his academic background and his accomplishments. Abhijit Ghorai, sir, is an experienced biopharmaceutical professional who had been involved in biopharma product development, quality control, operations, regulatory filing for the last 17 years at various capacities. His focus in biopharma research has been understanding the mechanism of action and functional characterization aspects of large molecules, biologics, biosimilars. He had worked at the R&D departments of Biocon, Dr. Reddy's, Lupin, Pfizer, etc. Currently, he works for Cognizant Life Science, where he focuses into digitalization areas within life science industry. Today, Mr. Abhijit Ghorai sir will be giving a presentation on the topic Overview of Biopharmaceutical Product Development and Commercialization. Now I would request Ms. Shalini Chaudhary from second year Department of Biotechnology to please come forward and felicitate our distinguished speaker.
Good afternoon, everyone. First of all, I would like to thank my respected teachers and the faculty members as I have been alumnus of this institute way back 2021-25 batch of BTech. I'd like to thank the organizing committee, and it's my pleasure to be part of this NHPT today over here. Would like to convey my regards, and I will start my talk on this. So I will be covering on the overview of biopharmaceutical product development and commercialization. So the way I've designed this slide, I would like to make it pretty interactive with the students, and so that we understand the flow of what happens in the biotechnology and biopharmaceutical industry, what are the different areas, and where actually students can have their potential opportunities for the career. That's the agenda of today's talk. Vis-a-vis, I will cover entire product development flow of the biopharmaceutical industry. All right. So the topics that will be, covering up with first, what is biopharmaceutical and what is a biotech drug, and then I will be going towards a little bit into how a biotech drug is developed, with the focus on the R&D and then the analytical characterization and the product life cycle. I'll share my experience. Before I start, I would like to hear from the students what you understand about a biopharmaceutical. All of you must have heard that, when we say a pharmaceutical drug or a chemistry drug, what comes in our mind? Apparently, first we talk about paracetamol, a medicine. Everyone are aware of that. When we talk about biotechnology drug or a biopharmaceutical drug, what comes in our mind? Anyone I will hear from the students? Any one of you? What is a drug which we call a biotech drug, which is not a classical chemistry oriented drug? Any one of you? Anyone from the students? No one? Sleeping? Any attempt? There is one of it behind. Please, go ahead.
You can get drugs from vegetables or pepper like capsaicin, which can be used to kill off bacteria like streptococcus, pneumonia, and other stuff.
All right, very close to that. So essentially, when we say biotech drug or biopharmaceutical drug, essentially something which is linked to the living organisms. It's not what the lab chemistry-driven drugs that are classically in market for many years. So this is something which has come from living organism. That's actually the biotech drug is all about. All right.
টানলে কি এটা খুলে যাবে? এটা ঘোরানো যাবে না।
All right. So biopharmaceuticals and the pharmaceuticals from biological sources. That's what the biotech drug is all about. This is one of the major area and in the biotechnology where biotechnological and biopharmaceutical drugs are dealt with. So different areas under this category is somebody mentioned about vaccines over there. Yes, vaccine comes under biopharmaceutical. Cell and gene therapy products comes under biopharmaceutical area. Novel biologics and the biosimilars. Novel biologics are the new drug discovered which is from the origin of the cell or biotechnology, and biosimilars are similar to the novel drugs. When the patents gets expired, people try to make a similar drug, that's called biosimilars. These are the major different areas under the biopharmaceutical category. Alright, so now I want to spend time on this entire biopharmaceutical drug development flow, where it actually starts from. So it actually originates from the discovery phase, where what we see on one slide over here is actually a phase of, on the case of new drug discovery, it could even go 15 to 20 years journey. Or in a biosimilar drug space, easily 7 to 10 years journey in the phase of the entire biopharmaceutical drug development space. So discovery stage starts where we have something at the inception level, where anything is found where something has a target which could have a disease target. That's where a lot of screening happens, and then a target is identified at the level of discovery. And then when the preclinical stage starts, where actually a lot of lab-based research work is being done, where to understand the structural understanding of the molecule, functional understanding of the molecule, how that can actually work in terms of the animal systems, in the in vivo conditions, in the lab scenario. A lot of safety studies, that's all actually happens in the preclinical phase under the product development. So now when we say preclinical, that means it is before the clinical trial. So that's where after you have a significant data package, what you go for is the IND, which is the new investigational new drug application, where actually you look for, you actually find that you are coming up with a new drug that you would like to further develop for the commercial purpose. So that's the stage. Now they enter into the clinical trials, where we have classical, there are phase 1, phase 2, phase 3, different stages of clinical trials, and then it goes for the review and the regulatory approval. So now these phase 1, phase 2, and phase 2 studies are the huge clinical trial studies which are done across these organizations when it comes for the discovery to the clinical trial, where a lot of patient data is generated through different studies, toxicity studies and other things. And then finally it goes for, you guys must have heard of the FDA, EMA, all these different regulators. That's where it goes for the regulatory approval application. Once this regulatory approval is done, and then it comes for the clinical launch of the drug, and then what goes for approval, something is called is MAA, Marketing Authorization Approval. That's where the drug has come up to the level of marketing, and then it goes for the post-approval studies. So this is a classical journey of the drug development flow. Just to get your students' interest, if you look at this entire flow, we have mentioned this discovery phase at the very beginning, and then you have mentioned the preclinical, where there are a lot of safety, efficacy studies and all. Then you have the lot of clinical trials which is involved. So now in the biotechnology, all of you actually in the college, what the subjects you study, they are all of this drug development flow. That is something for your information. If you see the discovery phase in the lab, you do a lot of biochemistry, immunology studies, a lot of animal cell culture lab, you also do RDNA technology, then everything comes at the discovery stage when you try to understand a molecule. Similarly, in the preclinical studies, there are different lab-based R&D research activities. That's where you guys actually do different kind of assays. Assays means you in the lab you must have done different kind of ELISA assays and seen different kind of chromatography assays. You must have heard of the HPLC assays and all those things. So all this come as a part of the preclinical research that you actually do in the discovery phase. You must have heard that clinical trials and this CRO, clinical research has been a big domain. You guys were asking some time back, so biotechnology, what all we can do? That's another area which is under clinical trials and the clinical research. That's another complete domain where you can get into with a biotechnology degree and the clinical trials area, understanding this clinical research, understanding pharmacovigilance, understanding these bioequivalence, BAB studies. There's an entire different opportunity in the biotechnology area of clinical research and then I guess you guys have heard of the medical writing, which is another new field. I have quite a few of our HIT pass-out students I have seen they are into medical kind of medical writing and all. That's linked to the regulatory filing and approval where you accumulate a lot of data, write a lot of documents and then organize them to the regulators. And then post-approval, what comes is the life cycle management. That's another complete management of the drug profile that happens in the entire business of drug. So this is a very brief about the drug development process. So now I will focus a little bit into the R&D part since I am from the discovery background. Before I get to, I would hear anyone has any question on this slide? Yes.
What kind of service will perform after you have launched the product?
After you have launched the product, there is something comes under the product lifecycle management. So in the product lifecycle management, the first study which is done is the trending of different parameters at the quality scale for the product behavior as well as the post-marketing pharmacovigilance at the drug safety level. So once you have launched, your R&D for the drug is mostly complete by default. The drug is at the commercial phase. That means now you have launched means you have to manage the supply to the market continuously. For that, your manufacturing facility should be on for that drug and your quality testing laboratory should be on. So now the drug when it is market, in the quality testing, a huge trending is actually behaving while it is going from the lab to the market. And pharmacovigilance is the another area which is post-marketing, which is done for to study the adverse reaction. There's a group of doctors who does it, who actually looks after any kind of adverse reactions reported for the drug or not. So these are the two major areas on the post-launch on the technical side. In addition, there is a lot of commercial sales-driven activity which happens for you have a complete strategy where you actually compete your certain market and then have a constant supply and get the business going. I have one slide on the product life cycle. There I will cover a bit on that. Any other question on this flow? Come on, students. Any other question? Okay, I'll come back to end again. Okay, so now getting into more for what happens actually in the drug development level. It's a classic example for a biosimilar product development flow. So you start with the clone development level. What happens is clone or cell line engineering level is the first step where you actually at the cell level, gene level, you do some transfection activity where you insert your actually the gene of interest where you want to have a desired protein coming out of it. That's the first phase of activity. A lot of molecular biology and immunological work is done at the level of clone development where you first express your gene of interest and start getting your molecule secreted and then you identify what is the right clone which will actually produce that drug in a smaller scale in that level. That's the very first phase of this drug biosimilar as a classic case of drug development. Next you have, once you have identified your particular clone, what is producing your drug, you would be interested to purify the drug because when your drug substance coming out, that solution will have a lot of other unwanted proteins with it. So that's where comes to the purification steps. It is very important. You guys all must have heard unit operations of chemical engineering. You guys must have been hearing, and then you also have in microbial fermentation technology. You guys have already read in the lab. So now, if you actually do is upstream level purifications and the different kind of and the small reactor waste, and then you have downstream purification, and then the small reactor when you get different kind of purified products, and then finally you get into your drug of interest, drug substance or the drug product. So these are actually the different departments who actually work as a big group on this entire process at the research level to have your molecule monoclone level to the drug substance and drug product level. Other important thing you do in parallel, which is the bioanalytical characterization. So whatever drug is coming out, let's say at the clone level or upstream purification or downstream or at the final at the drug product level, you need to first characterize that molecule and see whether structurally they meet the intended purpose, functionally whether they are active or not. That activity you call as an analytical or bioanalytical characterization, where you do a lot of cell line based assays to understand the biological activity of the drug, as well as you do a lot of HPLC based techniques, mass spec based techniques, a lot of other analytical methods to understand that whether what is the structure of the molecule. So that's the core R&D flow. At the level, at the R&D level, if you see one side is the process flow. From the clone, it moves from the upstream process, downstream process, then finally you get a drug product. On the other hand, you're characterizing it at the each stage. If you look at the bottom part, it's a schematic from one of the publication where you actually see it's just like a vial, and then you have a seed culture, which is a small scale level, reactor level, and then it's moving to the production level, and then it's a pilot plant production kind of thing, and then you have a solid or liquid, then you separate it, and then you have chromatographic purification, and then again you do this kind of separation, and that's where you reach to the intended drug substance or the drug product. So this is the R&D level activities across different departments. That's what happens in the biopharmaceutical setup. So I will go a little bit into all these departments to give you a feel of what all the different activities that happen. So now if you see, clone development is actually a process that I explained to you at the clone level, where when you are starting actually from the gene of interest, you are doing a recombinant vector transfection, and then if there is something called limit division. Basically the target over here is to reach to a single cell, and that the single cell needs to express the certain level of antibodies that you want to be expressed. So you try to reach a single cell level. There are different techniques to reach there. And then finally you start them when you have identified a single cell level which is actually preparing or making your desired protein. You identify them, and then you make further subculturing of that, and then you bank it, and then something called cell banking, where your desired cell population that you would be using to produce the drug in a larger scale, that you bank it, you make a small scale cell bank over here, you call it as a kind of a cell bank to start the final upstream process initiation from that cell. So that's what happens in the clone development area. The very first stage where you essentially establish your clone and establish your titer of the clone, what is giving you the desired product. After that, you get to the upstream and downstream purification process. So in both of these, the purpose is actually to get into a separated protein of interest, because when you actually start subculturing them in the reactor, you feed them with different kind of nutrients, with different kind of parameters, oxygen, whatever other nutrients you are feeding them in the culture. It starts producing your desired protein with that, because you actually start with the basically Chinese hamster ovarian cell lines. When you start injecting weight, and when you actually see, you get a different kind of proteins coming up which are not specific to the interest of gene that you want to actually derive. So that's where you do a lot of and as well as your analytical department keeps testing them in different kind of structural techniques and functional techniques to see whether they are actually making the for the intended purpose or not. In the downstream purification, there are different kind of purification techniques. Essentially, you do one way that you purify the protein to get your desired drug. At the same time, you have different HPLC techniques here to essentially characterize them. That's overliner, right, and the upstream and the downstream. And we process the way we talk about upstream is a little ahead in the bioreactor when their first purification happens, and downstream when you have collected and then you are doing further filtration and different kind of clearance to get your desired product. So now once you have your downstream purification done, then you finally get to your drug substance or the drug product, which is essentially a small scale prepared drug in the lab. Once this process is set, now you are ready to move to the pilot scale. What generally happens in industry is, first you have a lab scale activities process optimized when you see your desired protein which you are getting expressed, and then you transfer the entire technology from the lab to the pilot scale, where your reactor size could be up to 50 or 100 liter maybe. And then when you have optimized the process in the pilot, it's to the manufacturing scale, where it goes up to 1 to 2 lakh size fermenter. That's where the production facility, that's where it operates. So that's the flow, the entire process flow. Now coming to the little bit on the characterization flow versus process flow, to give you an idea. So process wise, we looked at from the cell culture, which is clone development, to we reach the drug substance or the drug product level. Whereas if you see, these are the different kind of product characteristics of these product development, to understand the structural and the functional aspects of this particular molecule. So if you see these kind of techniques, they are either linked to your lab, whatever you study in your lab, either they are different kind of immunological techniques, or it is linked to biochemistry, or it is linked to animal cell culture. In any one of these techniques that you all study as a part of your degree. Right, so now with this, I will go to the little bit more on the methods. So with one of you was discussing in the what happens in different kind of the phases of different kind of assays that move. So assays means basically, preclinical to the marketed product. If we see, there are different phases where we do different kind of testing. One way you have the process side, one way you have the analytical characterization. On the other side, if you see, there are the different kind of the characterization assays which are established across different phases of the product development, and they have different set parameters of the acceptance criteria. Some of them happens in the GLP space, some of them happens on the non-GLP space, and then it gets to the clinical. That's the another area that I already discussed a little before. So this is the overall flow that overall happens in the drug development space, from the clone level to the marketed product level. So a little bit overview on that. I think one of you were asking, what kind of testing happens in the QC lab, and when you have a product developed and it is moved from the R&D to pilot scale to the commercial space, that's where your quality control labs come into picture. So the first phase, what happens, you have the R&D labs when you have the molecule characterized and identified. Then when it goes to the manufacturing, you get the routine samples for testing in the quality control laboratory. So the major role for a quality control laboratory is the routine testing for the batch release, as well as they participate in the different kind of stability studies. And when a molecule is in the commercial space, you have a routine supply, you do a lot of trending of different parameters in the quality control laboratories. That's how the flow happens. Post that, it gets into the product life cycle management. Little bit more detail of the product development and the early to late space. So this RS is something called reference standard. Essentially, when I have been telling that all the time that we are analytically and functionally characterizing the molecule with the different stages. So there are some reference standards which are used across these different phases. So you must have heard this WHO and this different kind of these regulatory bodies, they also supply some reference standards of the drug that you actually compare across in your product development flow in this process as a part of your molecule moving from the inception to the late stage. Right there. So what happens in the commercial is linked to the question one of you has asked, what happens with post-commercialization? So commercialization itself is a big chapter. So now a classical drug development takes, let's say, if I take imagine 10 to 12 years of entire development, and then you have some level of data generated at the manufacturing level and some level of stability studies. So now you are good to go for filing it. So that's where your commercialization starts first. So you apply for marketing approval, you apply this data package to different regulatory bodies. For US, it is FDA, for Europe, it is EMA, for Japan, it is PMA. These are different regulatory bodies. When you submit your initial level of data package and tell them, look, we have a molecule, we have a new drug, we have a biotechnology drug which is efficient to treat certain disease, and it has sufficient safety data, sufficient efficacy data, and then this is good for human treatment. And you submit with the first phase 1, 2, and phase 3 some level of clinical trials data. And then what starts at the huge level of audit? You must have heard that some facility has got a big audit, some companies are shutting down their facilities because they could not pass the audit. That chapter starts from here. So what happens in some cases, due to the high pressure, work pressure or different reasons, you might have made a product but that may not have made the desired quality. So when I say desired quality, it might have some impurities into it, or it might have some sort of the practices that you have followed, they might not have been certainly quite compliant. You might have followed some practices which are not documented well in the laboratory. That's where the automation area is coming into a big role player, where you have completely digitized, so that you are, whatever experiments you are doing in the lab, automatically it gets to your laboratory system, so that even if you forget to capture anything on that day in the lab notebook, you will not even have that scope, everything is online. So all these are part of the quality requirement of the audits when these regulators comes for auditing these bodies, these organizations. Once your audit is clear, that's where you get something called MAA, Marketing Authorization Approval. So that's where your commercialization starts. You are ready to market it now. So that's why it says sales and marketing, regulatory, dossier submission and compliance, and then what comes the major role is medical affairs and marketing. Because medical affairs is quite related to the pharmacovigilance, which is the drug safety, how the drug is getting consumed or getting into the human body and how patients are behaving. In India, we generally don't look at it. Even if we see some reaction of a drug, we hardly report. In abroad, in Europe and in US, it's highly critical. Even if you have used an ointment and you have some itching, that gets reported. So that comes under this space of the medical affairs, and then market access is also a part of the marketing. It's very critical which market to sell. This is also linked to the patent, which molecule you have actually prepared and which market you can sell it. We are getting into all this comes into picture as a part of lunch and the commercialization. Yes.
Mr. Godoy, I will appreciate if you can finish in another 5 minutes. Actually, we have another session pending. So
you want me to cover it in the next conversation? You have 5 minutes? Perfect. All right. Okay, so now we'll get to this product lifecycle management is one more topic that we had one question. I've already briefed what product lifecycle is actually all about. So what I explained, that's one part of it. Other part of it, product lifecycle management is also a new area where a lot of different kind of techniques are being imparted to strategize and manage the product lifecycle, the entire flow, how the product is behaving from the start of the commercialization, and then what is the competition that comes under the product lifecycle management. Okay, so last but not the least, which is the timelines and the cost. So we say drug development, drug discovery, but which is actually an affair of huge cost. and use timeline. If you are working on a biological drugs from the scratch to the discovery to the commercialization, it could even take 15 to 20 years. Coming to the space of biosimilars means a new drug which is already there and you are copying or trying to make a similar drug to that and showing that it's comparable with innovative drug. It easily takes minimum 6 to 7 to up to 10, 11 years for a biosimilar drug coming into. So that's the space, a lot of automation driven applications including IoTs and other things coming into picture where we can bring this timeline down. So that's a huge area in the biotechnological field. How do we bring the time and cost down? That's where a lot of these automations and digital transformation space is coming into in this area. All right, so I have a last slide, which is just to show you how to grow a tree. You just get a seed into the soil, and then you water the seed, and then you see how the plant starts growing. But you cannot get the immediate fruit coming up out of it, right? It takes time. So that's exactly what happens in your process research, and then you have a formulation research, and then you have analytical research. And finally, when you are in the market of the drugs, then it's a billion-dollar business. So any biosimilar or biotech drug you take, they are all in the space of billion-dollar businesses. So that's where you actually start making revenue of the organizations, and then you are into the actual business. Okay, one more slide on the little bit to share my experience when I was working with Lupin. So this is a pharmaceutical company. I was in a biotech division of Lupin, which is in Pune. So I and myself and my team, we picked up a project which was at the late stage, and it was an anti-TNF molecule, etanercept, for the autoimmune disorder. So this, we were about to file that molecule in the regulatory bodies, and then we realized that the operations quality control, we had some challenges in the method, and it came back. So then we had to address all the challenges, and we had to rework on certain of the development and discovery aspects, even if that is not in the classic phase when the molecule is at the late stage, and you address it, and then you again go for the filing. And once you file any molecule, the first thing what you get is a big set of regulatory queries, and then your timeline starts over there. On these queries, you actually address the queries, you interact with the regulators, and then again you try to claim that your molecule is actually behaving well, that it is intended for the purpose. And then finally what you get is that regulatory approval. Is just like, wow, you have worked on some drugs, you are actually touching patients' lives. Essentially you are doing something novel for the society. The drugs that you are working on, they are getting to human body, your patients are getting cured. That's what happens when you have this. Couple of glimpses on the laboratory side, just for you to see how our industry lab looks like. So these are the different kind of characterization and cell culture laboratories. If you see, there are different blocks where the scientists are working. The bottom panels are the linked with the cell culture lab, where the middle panel is into your different kind of HPLC techniques, and the top panel is also different kind of HPLC and other techniques. Classical, how the lab looks like. I have another slide how manufacturing facilities are like. So if you see, these are all reactors or the fermenters of the different sizes. So this is how a classic manufacturing facility looks like in any biotech drug companies who are into this business. All right, so in the best interest of time, I'm going to go more detail, but to take questions, anything that comes to your mind, I want to hear from you guys.
Thank you, Mr. Roy, for your nice presentation. We can, I think, take one question quickly. And later on, if you have more questions, you can ask Mr. Roy. But right now, we are ready to take one question.
Any questions, anyone?
Yes, please.
We are testing in the lab, hardly two numbers comes in our hand and could be into the other thing. So, sir, don't you think that there should be, like, there's a huge gap between what we are doing in the computer aided part and what we are
Yes, so absolutely there are a lot of work has been done and going on. So computer-aided drug design and the doping-related areas and the drug design and drug discovery related areas using bioinformatics tools that I have not covered in the talk. So that comes in the phase of discovery where you have a screening. Let's say you want to screen 10,000 different leads and from there you want to bring down to 2 or 3 different leads. You cannot do that in the lab. That's where your drug designing and the docking stations, bioinformatics tools play a big role. There are a lot of work has already been done and going on. That's one area. Another area on the discovery phase and also in the, which is been not only discovery, in the routine preclinical phase also which is being done. Let's say you have one molecule X where it is already, you have developed it. Now you are developing molecule Y. What is the similarity of molecule X and Y? Whether these similarities would actually kind of replicate in the experimental data or not. So if it is yes, then can we bring the time down? Let's say you have done 15 different experiments for molecule X, can you do only 5 different experiments based on the experiments of your molecule X? Using a lot of automation as well as AI techniques, all these things are going on. To answer your question, this is already in place and a lot of things have been there, and if you see the drug discovery space, especially in the pharmacovigilance area and as well as the lab area, a lot of timelines have come down with the help of different kind of automations. One of them is artificial intelligence. It answers your question? Any other question? I'm here. After the talk, we can take offline questions.
Okay, thank you. Thank you, Mr. Goyal. Nice presentation.
Thank you.
Thank you so much, sir, for such an informative presentation and for taking time out of your busy schedule to come and share your knowledge with us. Now I would request Dr. Shudip Das, sir, Associate Professor, Department of Biotechnology, to felicitate our esteemed speaker with a token of appreciation. I would now request Dr. Shucheta Das Maji ma'am and Manushika ma'am to felicitate our Honorable Chair Sir, Dr. D.K. Jana sir and Dr. Shomitra Kaur sir. We'll shortly be beginning with our oral presentation session.
বসবো এর উপরেই বসবো এর উপরেই বসবো না বেঁচে দেয় ফোনটা নিতে ফোনটা দেখ কি করছিস কেন তো আমিও জানি কে পেছনে বাড়িস কেন আচ্ছা ওটাকে ভাই সত্যি পড়ে যাবে স্প্রিং ব্রিক একটু ক্যামেরাটা ঘোরাই আরো লোকজন গেলাম জুম করা যায় না?
হ্যাঁ দেখ, আরে ভাই চব্বিশ হালকা জুম কর।
দেখলি তোরও একই হলো। দাদা সামনে চলে আসলে।
We will now begin. This session will be chaired by Dr. Shubhit Das Sir, Associate Professor, Department of Biotechnology, Haldia Institute of Technology. And the session will be co-chaired by Dr. Mukesh Singh Sir, Associate Professor, Department of Biotechnology, Haldia Institute of Technology.
পিছনে ঘুরিয়ে দিবি।
কেন?
I request sirs to come on stage.
ও মানে এইটা
মাঝখানে পুরো একবার নিয়ে নেবে।
আরে জল পুরো পেছন হলতে পড়বো। এমনি পেছনে ওকে ডাকতে হবে, কে কে ঘুমাচ্ছে কে কে জেগে আছে।
আমি না বাড়ি চলে যাচ্ছি, করে 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 সবাইতেই আমাকে
The first presentation will be done by Ms. Shantanu Chaudhary from the School of Applied Science and Humanities of Haldia Institute of Technology. The title of the presentation is Understanding Protein Nanoparticles Interaction from Molecular Perspective. I request ma'am to come on stage. Also a note for the speakers, there will be 7 minutes for the presentation of the oral presentation and 3 minutes for interaction.
Today I am going to present my talk on the from molecular perspective. So the term nanotechnology is very close to the term biotechnology. In the recent days, there are many more researchers compiling the two things, nano biotechnology. So all of you know that there are many more application sites of such nanomaterials, say antimicrobial behavior and pathogenic behavior. There are applications in cosmetic and medical appliances, photoimaging, photothermal therapy, gene therapy, nanosensing, nanobiosensing, etc. Some of the things like photoimaging, photothermal therapy, magnetic responsive drug delivery, these things are already else. So, from this perspective, a nanoparticle can come into contact with the protein in many ways. The effect, therefore, of interaction between the proteins and nanoparticles is very necessary to understand. Because these tiny particles may affect the structure and function of the proteins, and also, whenever the nanoparticles is interacting with the proteins, the purpose of nanoparticle may also get hampered. Say, whenever the particle is attached to the proteins, maybe their targeting behavior is getting disturbed. So, by studying this protein nanoparticle interaction, we can understand the bioreactivity of nanoparticles, their usability, and also we can create a database which will be very helpful for nanomedicine, nanodiagnosis, and nanotherapeutic purpose. So there are different type of factors behind this protein nanoparticle interaction. What are the factors? There may be particle composition, there may be hydrophobicity, charge, size, surface property. All these things are very much responsible for this protein nanoparticle interaction. And the effect of this interaction is in numerous ways may come out as because of the surface contraction, there may be amyloid fibrillation, there may be oligomerization of the proteins, there may be oxidative damage, therefore the reduction of the enzymatic activity, etc. So, and also in another sense, whenever we can use the nanoparticles or nanostructures for any targeted purpose, what happens when nanoparticles be open to the cell cytosol? There are numerous types of proteins which get absorbed on the protein surface, thereby forming protein corona.
it may be a monolayer, it may be a multi-layer on the nanoparticle surface. Therefore, it loses its purpose, it loses its targeting behavior. So, what is our approach to study from two different aspects? What is the role of protein primary structure on the nanocluster property, and in vice versa, what is the effect of magnetite nanoparticles on the structure and function of protein? To study these two cases, we take a protein named MPT63. I am coming in later to that point. Before that, I just want to give all of you a very brief idea about the nanoclusters. So what are nanoparticles? According to its name and according to its dimension, is of size 2 nanometer, 200 nanometer, and more than that. But nanocluster is something which we often call a bridging link or a missing link between the molecules and particles of size less than 2 nanometer. And very interesting thing is that in these nanoclusters, there is discrete energy levels just like normal organic molecules in comparison to the bulk metals or bulk molecules or metal nanoparticles. So because of that, these metal nanoclusters shows very interesting features. They have some very beautiful bright fluorescence behavior, and depending on their energy gap, we can tune this energy gap. We get very high, very promising fluorescence characteristics. They have very high surface area, antielectrical property, energy band gap, electric properties, magnetic properties, etc. So, in context of the biological or biomedicinal field, the nanoclusters are very useful for applications. Not only the nanoclusters, actually we should tell this that protein or peptide conjugated or biomolecule conjugated nanoclusters are very useful for different type of biomedicinal purposes such as bioimaging, enzyme activity. Also, they can be made with chiral behavior for very selective binding or selective purpose. So, and our model protein is MPT63, which is a Mycobacterium tuberculosis derived protein and proposed as a TB antigen. We chose this protein because this is a very small protein and it has antigenic characteristics. Now, in the first case, we used this protein for the preparation of the gold nanocluster. Why we use the protein? Because for the preparation of gold nanocluster, we need a template, we need a stabilizing agent. Now, protein is a very good choice because it can behave as a stabilizer and also as a reducing agent. So, we didn't need any external reagent for preparing this gold nanocluster. Now, wild-type MPT63 didn't have any thiol residue, and thiol, that means SH, is very important for preparation of the gold nanocluster, since gold make a very strong bond with the sulfur atom. So we intentionally make mutation at 2 points, at point 20 and at point 40 to replace glycine to cysteine residue. Therefore, we intentionally introduce one thiol residue in the 2 mutant at 2 different points. So, in the primary structure, we find that there are 2 cysteine residue in the 2 mutant. Now, whenever we try to synthesize gold nanocluster by using these 2 mutant, mutant 1 and mutant 2, we find that in both cases we find 2 clusters of 5 atom and 8 atom, whereas for mutant 1, we find that 5 atom cluster is a major one, whereas for mutant 2, 8 atom cluster is a major one. And we differentiate this population with respect to their fluorescence intensity.
2 minutes are left for speaking. Please.
But fluorescence intensity is not the only parameter to differentiate them, so that some other experiments is also done here, such as XPS and TEMP to distinguish them. After that, some DFT calculations and simulations were also performed to support our experimental observation, which showed that actually there is what we observed in our experimental observation, that in for mutant 1, AU5 homo-orbitals is of lower energy, whereas for mutant 2, the AU8 cluster is of lower energy. In case 2, where we use magnetic nanoparticle for studying the effect of this particle on the protein, we find that there is a significant change in the wild type protein, and which we termed as conformational switch. This MPT63 sequence analysis showed that it has some chameleon sequence, and in presence of the nanoparticle, when it gets absorbed on the nanoparticle, in some chameleon sequence there is a conformational switch from the originally or native beta sheet to the alpha helix protein. So what we did, we again intentionally mutated that chameleon sequence patch to find out whether we can stop it or not. And it was observed by studying that different type of mutations, that for this tryptophan mutation, we found that after mutation, even if the protein while interacting with the nanoparticle, there is no change in its immunogenic activity, and protein showed its normal immunogenic response even after adsorption on the nanoparticle surface. So we can conclude with that prior to application of nano scale system, we need to understand the exact interaction pattern of the particle with the protein, and the change of and the effect of protein on the particle, so that we can design in the proper ways the particle or the nano system, the composite system for their effective behavior. I express my gratitude to Haldia Institute of Technology, my guide Professor Krishnananda Chakrabarty, my colleague Dr. Indra Londi, Dr. Achinta Shanigrai, and for fund DST Nanomission and DST NPT. Thank you all.
How you have made the conjugate, the protein conjugated with the nanoparticle?
We actually started.
Escape in the.
We actually started absorption. We didn't make any chemical.
You have conjugated the mycobacterium protein with the nanoparticle.
No, we didn't conjugate it. We simply add, because since there is ion, this SH group readily react with the gold. So it didn't need any external conjugation procedure.
Anyone from the audience?
Anybody who wish to question ma'am from the audience? No.
Any question?
Thank you.
Okay, thank you, ma'am.
Thank you, ma'am, for your presentation. Moving on. I would like to, ma'am, can you please wait? I would request Professor Shudipta sir, Associate Professor, Department of Biotechnology, to felicitate ma'am with a token of gratitude. The next presentation is by Mr. Ovirup Malla sir from the Department of Biophysics, Molecular Biology and Bioinformatics, University of Calcutta. The title of the presentation is Synergistic Anti-Cancer Effect of Combined Use of Dapagliflozin and Diclofenac Sodium against cervical and cancer cells. I request sir to please come up on stage and present.
তিনজন
Hello. So, hello everyone. Today I am going to present one part of my research work which is titled as Synergistic Anti-Cancer Effect of Combined Use of Dapagliflozin and Diclofenac Sodium against Cervical Cancer Cell. And I am Obhiru Molla and I am doing my PhD work under the guidance of Dr. Subhrama Gupta. So we all know that what is mitosis and cancer, because mitosis is a regulated process where a single cell divides into two identical daughter cells. And when a cell undergoes an unregulated division, cancer arises. And antimitotic agent is an agent that inhibits the cancer cell growth by inhibiting cell division. So basically diclofenac is basically our drug of interest, which is a member of non-steroidal anti-inflammatory drug that has been used for its analgesic, anti-inflammatory and antibiotic properties. And this figure shows the mechanistic action of diclofenac during inflammation. So diclofenac basically interferes in this pathway and inhibits the one major enzyme known as the cyclooxygenase 2. But there are reports suggest that besides anti-inflammatory functions of diclofenac, there are anti-cancer properties also. So we have found some preliminary results. So at first we are trying to find out what is the cytotoxic effect of diclofenac in cancer cell line, and we use the HeLa cell line, that is the cervical cancer cell line, and it has been observed that diclofenac dose dependently and time dependently inhibits the cell viability, and it also increases the caspase-3 activity, which is a marker of apoptosis. And from this data, we can say that diclofenac induces apoptosis-mediated cell death in cancer cells. However, it does not have any significant effect on the non-cancer cell line. From this, we can say that cytotoxic specificity of diclofenac is very much cancer cell specific. And from literature, it has been shown that diclofenac shown its anti-cancer property by interfering in glucose metabolism pathway. But its molecular level target has not been identified. So from this, we predicted that any glycolytic enzymes can be a probable target. So for this, we used a molecular docking analysis approach, and we took all the crystal structure of glycolytic enzymes and used molecular docking analysis and calculate the docking score. And from this, it has been observed that among all glycolytic enzyme diclofenac complex, lactate dehydrogenase A shows the least docking score, that means the more favorable binding, which ensures that lactate dehydrogenase A can be a probable target of diclofenac. So what is lactate dehydrogenase A? So lactate dehydrogenase A is basically mostly expressed in cancer cell due to the instant energy production, and it is responsible for this Warburg effect, and this is important for this conversion of pyruvate to lactate using NADH as a cofactor. And to check whether our in silico data is correct or not, we check the expression of activity of LDHA in both cancer cell line as well as non-cancer cell line with different concentration of diclofenac, and it has been observed that diclofenac inhibits the lactate dehydrogenase activity in cancer cell line, and it also inhibits the pure LDHA level, and it increases the substrate level, which indirectly entails that diclofenac inhibits the LDHA. And these are the probable downstream factors if LDHA is inhibited by diclofenac. So let's say this is LDHA and there is no diclofenac, so glucose uptake is higher, and as the substrate is converted into product, all the product level is increased and the substrate level is decreased. But if diclofenac inhibits the enzyme, the glucose uptake will be lower and the substrate gets accumulated, for this substrate level is higher and all the product level will be decreased. So to check this whether this happens in cellular context or not, we measure all the metabolites level and with different concentration and time point of diclofenac, and it has been observed that diclofenac elevates the pyruvate level and it also decreases the glucose uptake, NAD+ level, and lactate level and intercellular ATP level, which suggests that diclofenac is an inhibitor of lactate dehydrogenase A. So now an obvious question arises that whether diclofenac shows any synergistic effect when it is combined with the type 2 sodium glucose cotransporter inhibitor. So let me introduce you some brief information regarding this type 2 sodium glucose cotransporter inhibitor. So they are basically an anti-hyperglycemic agent that is mainly expressed in basically renal proximal convoluted tubule. And it exerts its effect by preventing the reabsorption of filtered glucose from the tubular lumen. And dapagliflozin is one of the members of this type 2 sodium glucose transporter inhibitor. So this figure shows that the mechanistic action of this dapagliflozin during hyperglycemia. So in normal physiological condition, so the most of the glucose is reabsorbed from this nephron and returns back into the blood. As a result, there will be minimum secretion of glucose to urine. But in case of hyperglycemic condition, when this dapagliflozin has been treated, so most of the glucose remains in the renal tubule, and as a result, most of the glucose will be secreted through the urine. So in this way, dapagliflozin inhibits these hyperglycemic condition. So we implemented this idea in our work. So let's say this is the cell, and this is the extracellular part, and this is the cytoplasmic part, and this is the glucose. This glucose is quickly uptake by this sodium glucose cotransporter and utilized and formed pyruvic acid using with this process known as the glycolysis, and this via lactate dehydrogenase A, it is converted to lactic acid, and which is the extracellular acidification, as well as it produces ATP, which is important for cellular energy source. As we have stated earlier, that diclofenac is an inhibitor of LDHA. So it reduces the ATP level and lactate level. And the theory is, if dapagliflozin inhibits more glucose uptake, can we get any therapeutic approach or not? So to answer this question, we at first calculate the IC50 value of dapagliflozin and diclofenac in HeLa cell line, and it has been observed that both dapagliflozin and diclofenac reduces the cell viability in a dose-dependent manner. Now, we check the combination effect of dapagliflozin and diclofenac cell viability assay, and it has been observed that both the treated combination treatment showed the more synergistic effect compared to the single treatment. Now, a question arises whether this combination treatment is synergistic or not, and for this we use an widely used equation known as the Chow-Talala method, and which calculates the CI score. And when I fit this data into this equation, we found that the d2 by dx2 value is 0.212 and d1 by dx1 value is 0.1. So from this equation, if this CI score is less than 1, that means the effect is synergistic. So for this case, our case, we have observed that the CI score is 0.2 plus 0.1, roughly 0.3. That means the effect is synergistic. And now we want to check whether diclofenac and dapagliflozin synergistically induce apoptosis or not. And from this data, it has been observed that diclofenac and dapagliflozin synergistically induces apoptosis compared to single treatment. And it both synergistically inhibit LDH activity, but dapagliflozin does not have any effect on pure LDH activity. From this, it can be concluded that these two drugs operate two different mechanisms. And now we want to check whether the dapagliflozin and diclofenac work synergistically affect on glucose uptake, extracellular acidification, and ATP level or not. And it has been observed that in all three cases, diclofenac and dapagliflozin shows the synergistic effect. So in conclusion, we can conclude that diclofenac induces apoptosis in cervical cancer cell in dose and time dependent manner. Diclofenac does not show any significant cytotoxic effect in non-cancer cell line, and diclofenac inhibits intracellular LDHA activity in cancer cell, and diclofenac shows great synergistic effect in combination with dapagliflozin. So I want to thank Dr. Suprema Gupta and Unasu for their valuable guidance. So the Institute of Technology and University of Calcutta, Department of Microbiology, University of Calcutta for Docking Studies, and the funding agency. Thank you.
So this is the special thought question for any question from audience.
The guide itself?
No. So there is no effect of DAPA that you have shown now. Sorry, DAPA. Yes. So DAPA, there is no effect in
Actually, data deposit and
So you go to the conclusion first.
So here they use DCA.
Yes.
So what is the function of DAPA here?
DAPA actually is the glucose transport inhibitor. So actually it inhibits the glucose uptake.
So we have to administer both the drug.
Exactly, combined drug. And at first, dapagliflozin performs the inhibition of the glucose from the glucose source, and diclofenac basically inhibits the glucose utilization. So in both these cases, we get synergistic effect. So actually, DAPA inhibits in this glucose uptake, and diclofenac is inhibits this pyruvate to this lactate production. So if we use this both of these drugs, actually the IC50 concentration is significantly lower.
Thank you. Now I will request Dr. Mukesh Singh sir to please come forward and hand over the certificate of participation to Mr. Ovirup Malla. Thank you, sir. Next we have Shravanti Gayan coming from the Department of Physics, Muishadal College, Purvo Mednipur, and she will be presenting on the topic tuning the luminescent properties of fluorescent quantum dots.
ডিপার্টমেন্টে এমনি একদিন করতে হবে এখন পাচ্ছি না করতে আমাদের তাহলে আমি দিতে পারি আমার তোমার মেন্টাল প্রিপারেশন নেই না আমি আমার রেকর্ড আছে মেন্টাল প্রিপারেশন না
Good afternoon to all. I am Shravanti Gayan from Department of Physics, Muishadal College, Purvo Mednipur, and School of Applied Sciences and Humanities, the Institute of Technology.
Madam.
The title of my oral presentation is Tuning the Luminescence Properties of Fluorescent Quantum Dots.
The outlines of my presentation is to design such a quantum dot, synthesis of it, characterization, and applications of it. For this, to make a quantum dot, we used in our lab water in oil microemulsion method. Here we take sources of manganese, sulfur, zinc, and cadmium in different proportions in different containers, and then make a aqueous solution of it and put it in a oil solution. So after that, by magnetic stirring, we got the surfactant molecules only. This is basically a robust technique for producing monodisperse nanoparticles at room temperature. One advantage of this method is that particle size can be tuned by varying microemulsion parameters, and a wide variety of wide water-based chemistry can be performed in this nanoreactor. So basically what we found, a nanoscale reactor for nanoparticle cells. This is the arrangement diagram for fluorescent quantum dots. First one is for intrinsic semiconductor quantum dots with banded emission, and second one is for dopant-based semiconductor quantum dots with banded emission. Here we used the second one. Here an additional band can be seen within the conduction band and valence band of the host. This is basically dopant ion energy level that serves as the additional luminescent centers. This is the plot where we have shown surface defects and corresponding surface states that can influence the luminescence characteristics of the quantum dots. Here surface states are seen, and we try to minimize these surface states as it acts as a non-radiative relaxation center by trapping electrons and holes. For passivation, we use inorganic passivation as it is quite stable. It can enhance quantum efficiency and improve photostability also. This is the schematic for photostability experiment of quantum dots with and without Jeter scattering. In the first plot, we have seen that when we used CDS core, manganese to CDS core only, then the emitted intensity has very low value, but when we used manganese to CDS core with ZNS cell, then emitted radiation has a very large intensity. And in the right side, we have shown here inorganic passivation in the upper half and organic passivation in the lower half. In organic passivation, its stability is very good, but organic passivation is just degrading. So we use inorganic passivation, it is highly stable, and it is ideal for real-time biological experiments like stem cell tracker, cell particle interaction, then value. This is the characteristics of manganese drop CDS core with ZNS cell quantum dots that we synthesized in our laboratory. Here, site and total emissions properties are shown. First one is the TEM image. From here, we got the diameter of produced quantum dots, and diameter is about 3.1 nanometer. And in the right side, we show PL excitation and emission. First one is for PL excitation, and in exciting line, there is almost a peak about 350 nanometer, and in PL emission, there is an emission at almost 590 nanometer in the inside. I have shown here a plain gravel of this quantum dot, and it is fluorescent, it is emitting yellow-orange emission at visible range. This is basically band gap engineering of CDS and ZNS. Here in dark case, for CDS, band gap is 2.4 eV, and for ZNS it is 3.6 eV. We try to make an alloy of these two, and it has this alloy has a band within these two, and a non-radiative decay occurs to the manganese D levels, and then 41 to 6A1 is a well-known levels of manganese, and this emission is about 590 nanometer, and yellow-orange emission occurs at this wavelength. This is the PL spectra of the alloy manganese doped CDX Z1-6 CDS core and with ZNS say quantum dot. Here excitation wavelength at a wide range 300 to 500 nanometer. When we use only ZNS code, the graph is given in the extreme left side, and when we use only CDS code, the graph is given in the right side. This is the Kell spectra of the quantum dot, alloy made quantum dot. Here from the graph it is clear that we have made different proportions of this core, which is made of alloy, and when the Zn is there, a little bit amount greater than CD, then we get maximum radiated intensity. So the summary of my work is highly stable, fluorescent, monodisperse, quantum dots are synthesized by the engineering through semiconductor preparation, and quantum dots with wide absorption in the visible range suitable candidate is a suitable candidate for solar absorber layer. Surface modification is used in chemistry for bioconjugation and introduction for other modalities. That's all about my work. I would like to thank my guide, Dr. Shomitra Kaur, Department of Applied Sciences and Humanities, HI. Thank you to all.
Okay, madam. What is your practice in integration or application of quantum dots? Is of biology or
First one, it is and fluorescent, so it can use many. We can use it in where fluorescence is necessary, and there are some other applications of bioimaging and brain cell mapping, other anything.
What is the property of being further? What property? Why it is further able? Why did it bring that?
Thank you, ma'am, for this nice presentation. I would now request Professor Shubhit Das sir to felicitate ma'am with a token of appreciation. Our next speaker for this event is Mr. Shukhan Das from the Department of Chemical Engineering, National Institute of Technology, Durgapur. The title of the presentation is Development of a Cost-Effective Microalgal Bioreactor for the Assimilation of Dissolved Inorganic Carbon, Enhancement of Biomass Productivity.
Good afternoon to all of you. Good afternoon, sir. Okay, thank you. So, good afternoon, sir. My topic is development of a cost-effective microbial bioreactor for the assimilation of dissolved inorganic carbon, enhancement of biomass productivity with essential bioderivatives. So, this is the index. Greenhouse gas. So, this is a great threat for the atmosphere, the climate change presently. So, now I will discuss with the different CO2 capture process and objective of my study: green environment, biological sequestration, collections, isolation, purification, and culture of microalgae. Then characterizations followed by different biomolecules. Then optimizations by Tagogi method. Then economics part and future perspective part, and definitely biodiesel. that would be a viable option for future. So these are the topic I'm going to cover. So this is the plot, this is the greenhouse gas emissions figure. So presently around 38 gigaton greenhouse gas is emitted per year. So out of this 38 gigaton, 21 we can able to sequestrate by land and ocean. So 17 gigaton is excess to atmosphere. So this greenhouse gas, especially this carbon dioxide, is accumulating in the atmosphere and causing this rise of CO2 ppm in the atmosphere and causing the climate change because of the rise in temperature. So there are different greenhouse gases are there. So contribution of CO2 is basically 77% out of this. So you know CO2 is required because we cannot live without CO2 because this is essential for survival of the autotrophs and the existence of the organic world because it captures the heat and maintain the earth temperature. So these are the different statistics. It shows that CO2 having different emission potentials per capita per annum. So India's position is 0.93 ton presently because if we compare, there's a comparison between developed country and the developing country. For the developed country it is very high and for the developing country it is very less. So this is the part is the positive side, but since the climate is overall integrated, so we cannot separate India as a whole because it is total wise the ppm of the CO2 that is entering into atmosphere and it is increasing and causing this climate change. So there are different steps that has been taken worldwide, like Kyoto Protocol, Paris Agreement, IMDC. I'm not going into detail because scarcity of time. And United Union, they have Europe Union, they have declared to abandon this hydrocarbon fuel by 2030. The Indian government also declares this net zero emissions by 2070. So these are the different agreements, protocols that has been taken following these reductions of this greenhouse gas by 2070. So these are the different technologies. So technology for CO2 capture, basically there are two processes. One is chemical process and another is the biological process. Chemical process having lot many disadvantages, cost, complexity, everything. There is this innovative toxicity is there. And also if we consider this biological process, that CO2 has to dissolve in water, but this solubility is very less because CO2 is staying soluble in water. So in any case, you have to go for the mineralization of carbon dioxide. This is a global discussion topic now because to combat CO2, you need some minerals. That minerals can be uptaken by algae and different microorganisms by their photosynthetic mechanisms to convert to glucose. From that, by different metabolic pathway, we can produce lipid and protein and other biomolecules. So these are the threat to the atmosphere. So these are the different threats are there. So basically the area of work is the green environment, basically the sample collections of the microalgae, these characterizations, lab scale productions of biomass is optimized cost effective way, and enhancement of mass production, extraction of value added products that I am presently working on. This is the sample collection site. This is in Haldia township, close to this confluence of the Haldia river. So sample has been collected and it has been inoculated into BG11 medium and where it is being cultured in the ambient conditions. These are the different plate splitting methods. You know that because by this quadrant splitting, we have isolated this single colony that has been cultured in agar and as well as this liquid medium. This is the characterization process. So this characterization has been done by 18S RNA. This is a method to characterization of this algae. So by this is the stain. This name is Desmodius intermediate. They are working on this. So these are the different lab setup. You can consider like variation of pH, temperature, light duration, light intensity, inoculum size, substrate concentration, nutrient agitations. So lot many parameters we have tried to vary and optimize these parameters. So based on that, we have some setup of the biological bioreactor. So this setup is available in this college in biotechnology department. So in this, you can see that this picture and this picture, the difference is the growth is clear. You can see from this green color. So from that, we have analyzed different components. These carbohydrate, protein, fatty acid, pigments, and these are the findings. So initially, it was for 2 gram per liter sodium bicarbonate. The biomass concentration was around 2.54, which is good enough biomass we have got. And this photosynthetic pigment also we have analyzed. So we have found out this 2 gram per liter was the optimized figure where we have received this chlorophyll A, B, and total pigments. Total carbohydrate and sodium also analysis has been done. So these are the protocol that is written in the right side that has been followed. And if you see this lipid analysis also, so this is basically 2 gram per liter, we have got around 27 milligram per liter lipid. So this is a establishing method to optimize, this is a statistical method by the help of S by N ratio, the signal to noise ratio, you can optimize given parameters. So we have found out certain value of pH, temperature, bicarbonate concentration. So based on that, this optimization has been done. So final target definitely to biomass, higher biomass productions and higher biomolecules, and from there we can go for this energy part, because biodiesel, whatever liquid is there in the biomass, that it can be converted into biodiesel by transesterification process. And if it is ethanol content is high, like carbon content, can go for bioethanol, hydrogen also. So different perspectives are there of this biomass. So this is the economics part. So if you consider, I'm not going into detail, if you consider this green part, that is $114 per megawatt of power generation, that is the cost for biomass energy presently. It is high compared to other renewable. If you consider solar, wind and hydro, geothermal, it is comparatively less. But still there is the options that we have to go for this biofuel, because this is only one of the options. And if you see the right side block, that why algae, because this is a statistics that is taken from Wikipedia. It tells very clearly if the total requirement of the petroleum fuel in USA, that can be substituted by algae. So only 0.42% of the US map, that land is required. It's very minimal, and you have to go for this process only, because this is the future and we have to survive without this petroleum fuel and the fossil fuel. So advantage of the biodiesel and disadvantage is everything is there. So definitely advantage more compared to disadvantage, but only one thing is that there is a cost. That cost optimization you are doing by the help of some biovector modeling and design. So this part I am now working on. And algae is the fastest growing plant in the universe. So that is also additional advantage to us. So we can have a rapid biomass productions and also the essential bioderivatives. So these are the things, these are the list of reference that have been followed during my study. And thank you very much. Thank you. Thank you. Yes, sir.
How do you
Yes, sir. It has been done by plate tracking, sir. Basically, we have followed this quadrant tracking. My dad single colony has been isolated. Then it is the universal medium in BG level. We have culture of that. By plate tracking, sir. Yes, sir. I did this purification for this culture. Initially, it was mix of multiple other algae. We have gone for this purification and isolation of the single colony. Then in solid and liquid media culture has been done. Thank you, sir. Thank you, sir. Thank you, sir.
Thank you so much. I would now request Mukesh Singh sir to please come forward and hand over the certificate of participation to our scholar. Next we have Professor Ranjay Kumar Thakur sir from the Department of Food Technology, Haldia Institute of Technology, and he will be presenting on the topic Novel Applications of Bioactive Elements in Aqueous Extract from Fruit Waste as Edible Packaging Material, a value-added food product.
না টপিকের উপর আমরা হচ্ছে বায়োবেসিস
মানে কোশ্চেনটা লিগাল ছিল
হ্যাঁ যে এখন ব্যাকটেরিয়া দিই তো ওয়েস্ট ওয়াটার সাথে রেমিডিয়েশন করি সুতরাং এবার বলেছি যখন ব্যাকটেরিয়াগুলো করবে ব্যাকটেরিয়াগুলো এরপর হয়ে যাওয়ার পর কী করে তুমি বার করবে
এই প্রেজেন্টেশনের কথা বলতে এইটার কথা যেটা আমি সত্যি করে ফাইনালি হচ্ছে হচ্ছে
আজকে ক্যামেরা নেই আরে ভাই ব্যাপারটা হচ্ছে বোঝার আছে বড় কথাটা পরের কথা আমরা বেঁচে আছে মালটা
25% আছে
পরবর্তীকালে যেগুলো আমরাই
Very good evening to all of you. Today I am going to give presentation of novel application of bioactive elements in aqueous extract from food waste as edible packaging materials. Value added food products. Now, as we know that nowadays waste management is very difficult task, and that waste cause various threat to our environment, and ultimately that affect the human health. And major part of that waste is mainly constitute of organic waste, mainly that are present in municipal solid waste, and most of that one are is actually the food processing industries which handle with the fruits and vegetables that actually increasing the concentration amount, and mainly fruit like solid fruits and fruit juices processing industries is throwing various fruit peels and various waste materials which comes as a municipal waste. And as we know that contains lots of bioactive elements, phytochemicals, which are actually most useful in if you consider pharmaceutical, cosmetic industries or food industry, that can be that has wide application of that phytochemicals. So we, along with my the mentor, think about how that why not we recycle? Why not we recycle that phytochemicals so that we can use that one, reuse that one for processing of different things. So for that we set objectives, and our objectives is actually phytochemicals profiling in fruit residual aqueous extract by TCMS, and thereafter evaluation of antibacterial antioxidants. Actually, the food waste bioactive elements mainly that act as an antioxidant and that has various antimicrobial activities. After that, as we target is to use that one as edible packaging material, so it must be necessary that that component must be biocompatible with the consumer. So we have done various in vitro biocompatibility tests, mainly of aqueous extract, and then we prepared formulate coatings for fruits and vegetables to see the shelf life, how can that increase or decrease. And then we thought about that nano encapsulation of that one for also to increase the produce the value added product. Now this is my outline of work. We first collected different fruit peels or fruit waste from the inorganic idea, and then that converting that one into product by mechanically after sun drying. After that, we have studied various preliminary phytochemicals present or not, qualitative and quantitative both, and then we profiling that chemicals by GCMS analysis. And after GCMS, we prepare different extract by using the 5 kind of the solvent like water, ethanol, petroleum ether and butanol. And then we prepare a thin film and apply that film on the food materials like this one, tomato and grapes, and we observe the changes. So it has seen that after 10 days, the fruits of these two fruits, tomatoes and grapes, shows a minimum spoilage in compared to non-coated one. So that will be discussed later. So we have done different studies and hemolytic test we have done. So our work plan is first we collected fruit from different areas, then phytochemical characterization of fruit waste, then qualitative and quantitative both. We determine different physical components like moisture content, volatile matter, fat, other. And this is the same study for microstructure analysis of powder, which shows various microporous, this one, and heterogeneous and microporous molecules is present, and that also contain different minerals also, carbon, nitrogen, potassium, magnesium that are present by that is actually we observe after the EDX studies of that powder, and that indicates this presence of different important this one. And after preparation of powder, we prepared extract by using different solvent. Actually, solvent extraction we have used by using the substrate method. This is the general procedure that we follow. Then phytochemical screening of extract, preliminary phytochemicals that are qualitative as we say. That shows different kinds of amino acid, estraconol, phytosterol, tannins. Besides that, we determine total phenol content, protein content, carbohydrate content also quantitatively. Then we send the extract to the for GCMS analysis, and we observe that there is a various kind of the biomolecules, bioactive molecules we found in the GCMS analysis, which plays important role and which are shown actually the antioxidant activity and antimicrobial in nature. So antioxidant means that prevent the oxidation, so preventing the spoilage by restricting the rancidity, and antimicrobial studies we also done studies. So different kind of this one. Then antimicrobial testing we have done by using the standard method. Then biocompatibility. That biocompatibility, two kinds of the experiment we have done. That aqueous extract on goats erythrocytes and observe the hemolysis, and we observe that compared to negative control, that shows minimum inhibition to the hemoglobin, minimum breakdown of the hemoglobin, so that so we considering that are biocompatible, the consumer can be easily used. Then we studied that one as the calf farmers DNA and fruit residue, calf farmers that DNA damage causing or not. Again, that giving the positive result. There is no more DNA damage, so that biocompatible again. Then after studying all that things, we are going to prepare, we getting converting that one in edible form. For that purpose, we converting that one in the edible package coating of the fruits and vegetables by picogen-based formulation we have done. General formulation we observe. We have done the coating, and in coating, after coating, we studied the various characteristics of the film also. We observed the same result of that one, and then we find out that there is a various kind of the essential minerals, micronutrients, and that are present in the found in the our this one thin film which developed by the picogen, and after that we see the application, and this figure shows that. So this first one, this part, this shows the figure of the this tomato and the grapes at 0 days. This when we purchase just after purchasing that one from the market, washing with the deionized water, and then we see the picture. After 10 days, this one is the fruit tomatoes and grapes which is coated with by the ketogen 3 film along with our fruit extract, and it shows the minimum spoilage we have observed here, while the uncoated one shows this one maximum spoilage. So these differences indicates that our extract if we use that one as a packaging material that increase the shelf life of the product, we can keep that one for longer time. This study is also indicate microbial, that means this first one is the this uncoated one. Second one shows the growth of microorganism after 10 days of this one, and this third one indicates those which are uncoated. So by that way we also study that besides the shelf life, that also inhibit the growth of microorganisms, so that increase the ultimately shelf life of the product. So work is going on, and this work is possible due to the support of from my guide, Dr. Bhupesh Singh, Department of Biotechnology, and Dr. Prashanto Kumar Vishwas, Department of Food Technology and Biochemical Engineering, Jadavpur University. And this is the publication we have done on our
সিভিতে নিশ্চয়ই ঝাপা সার্টিফিকেট দিতে পারবো হ্যাঁ কেন তুই
আরে সিভিতে মানে কি ওরকম সার্টিফিকেট অ্যাটাচ করতে হবে না, লিখতে হবে শুধু যে এই সার্টিফিকেট আছে আমার কাছে।
Thank you, sir, for this wonderful presentation. I would now request Dr. Swati Maiti ma'am to please come up on stage and present the certificate of participation to his respected sir.
কখনো একটু পার্টিসিপেশন দিচ্ছে এগুলো কীরকম সার্টিফিকেট পার্টিসিপেশন তো বলছি একটু অংশগ্রহণ
পার্টিসিপেশন সার্টিফিকেট রেখে দেবে, থই না কম্পিটিশন
না আর কি আর সেই গ্র্যাটিটিউড
আরে ওইটাই একই, যে একই রকম একই টাইমে বলে গেলে তো লোকেরা এগোই যাবে, তাই না? নানা রকম সিনোনিম অ্যান্টোনিম এগুলো জানিস, ওইটাই
গ্র্যাটিটিউড পার্টিসিপেশন একই স্যার,
একই জিনিসকেই ইন্ডিকেট করছে, সিনোনিম না
সার্টিফিকেটগুলো একই দিতে হবে।
I will now request Dr. Mukesh Singh sir to felicitate Atanu Bera sir with the certificate of participation.
তোমারও প্রেজেন্টেশন ছিল আমার 5 তারিখ
আমার অনলাইন হয়ে গেছে।
অনলাইন
আমাদেরও অনলাইন হয়েছে, ফোর্থ ইয়ারেরও অনলাইন, না আমাদের সেকেন্ড ইয়ারটা অনলাইন হচ্ছে, ফোর্থ ইয়ারের যে ডিপার্টমেন্টে অফলাইন হচ্ছে, ওদের তো আলাদাই হচ্ছে, পুরো এসএল হচ্ছে
আজকে আলাদা হচ্ছে না।
I will now request Dr. Kyashaw ma'am to please come forward and felicitate the chairperson and co-chairperson of this session, Professor Shudip Das sir and Professor Mukesh Singh sir, and felicitate them with the certificate.
নেক্সট এইচওডি ম্যাম আমাদের এই বছর তুই লাস্ট আমাদের ওরা এবার এনাকে ফেস করবি, ভালোভাবে করিস।
ঠিকঠাক আছে যে ক্যাশ না ওই চাপে সুপ্রভাত তো শুনেছি ক্যাশ।
We will now be continuing with the poster presentation session of the first year students. I will now once again request Dr. Rabindranath Jana sir to preside as the judge for this event. Once again, I would remind the participants that the time for presentation of poster is 5 minutes. The first poster for today's presentation session is from Polo Mishen, Protasha Das, Sreyoshi Kaur and Rajdeep Bose on the topic nanotechnology for treating cancer cells. I'm very sorry. The first presentation will be done by Vimasha De, Sampurna Shaha, Shubhashri Patro and Supriya on the topic improvisation of brain through BCR.
ট্রেনিং কিন্তু ভালো ভালো সাবজেক্ট আমি যদি টেনটা মারতে যদি তোর ফোরে না সরি আইটি ফোরে থাকবে তো সেগুলো বলছো হ্যাঁ ওই আমার আসলে ওই অ্যাডভান্টেজ সিফোটা তবে সিফোর সেটাই
কলেজে বাইরেও
করতে দেন না
Good evening, respected judges, professors, seniors, and my fellow batchmates. Our topic for today's presentation is improvisation of brain through BCI. My name is Vivasha Dey from Biotechnology first year, and I would like to introduce everyone to our team, Sampurna Saha, Supriya. So come to the next point. What is the history of BCI? The research on the BCI began in the 1970s in California with experimental performed on animals to develop a new dialect and the brain. In 1973, Jacques Vidal published a paper titled Toward Direct Brain Computer Communications. So what is the object of BCI? The main goal of BCI is restore useful function to people disabled by neuromuscular disorder such as cerebral palsy, stroke, and or spinal cord injury. So next point is what is BCI? BCI is also known as brain computing interface or smart brain, acquires brain signal, analyze them and transmit them into commands that are relayed to a output device that carry out desired action. I would like to my co-teammate to continue the presentations. Sampurna Shah.
Good evening, everyone. Now I'm going to present the advantages of brain-computer interface. BCI allows pupils to control machines using their thoughts. BCI will help to creating a direct communication pathway between the humans and the computer. It has a lot of advantages in the medical field for the disabled persons. For example, it allows the paralyzed people to regain their control over their limbs. For blind people, it transmits visual images so that they are able to see. The other fields like the gaming fields, it has many advantages over. It has many advantages for the gaming purpose. And for the disadvantage, I will say research is still in under process, and for the other one, I will say it's a very expensive. So it takes a lot of money for setting up the BCI environment. Rest of the part, I would like to call Subhoshri. So, moving on with the presentation, talking about the applications of BCI. It can be help to motor rehabilitation following psychiatric disorders. The main applications of BCI technology have been in the domain rehabilitation control with prosthetics and neurofeedback. Now coming to the future prospects, BCI technology is no doubt a growing field of interest. It has medical applications ranging from prevention, detection and diagnosis. BCI research is being done to develop devices that aid people with disabilities, especially those who are affected by neurological and neuromuscular conditions like brain and spinal cord injury. So, now we are at the end of our presentation. BCI has proved to be successful in its field, but still research is going on to improve and help a larger number of people. Thank you.
Session is open for
anyone from the audience who wants to answer or question?
তুই পারবি এই টপিকটা এটা এমন আনকমন টপিক আমারই তো টপিকটা দেওয়া হলে আমি তো পুরো জিনিসটা বুঝিয়ে দিয়েছি আমার থেকে তো নোটস নেই ব্রেন কম্পিউটার ইন্টারফেস আসলে ব্যাপারটা হচ্ছে এটা পুরোপুরি আইওটি আর আর্টিফিশিয়াল ইন্টেলিজেন্স বেসড ঠিক আছে তোর নিউরন জানিস
What are the different types of attention
নিউরনটা হেঁটেছে
to be used for this
ব্রেন কন্ট্রোল ইউনিট বলে যে কি করতে যেমন বড় জিনিস করে লাগিয়েছে সিগন্যাল না ব্রেন হচ্ছে এটাকে সলার থেকে তো এটাই হচ্ছে জিনিসটা তো এটাই হচ্ছে এবার যদি প্যারালাইজ থাকে
Thank you for this presentation.
কাজ করছে না
The next presentation will be done by Shruti Manna, Meghananda Gayan, Disha Dishra on the topic Effect of Polymer Type on the Colonization of Plastic Pellets by Marine Bacteria.
এটাই হচ্ছে বেশি আর্টিফিশিয়াল ইন্টেলিজেন্স
আবার যেমন নর্মাল মানুষ কাজ করে তেমন
তোরা তো চারজন তো বলিস নি মনে নেই
চারজনই তুমি কি ঘুমোচ্ছিলে
কথা বলছি না
কেমন হয়েছে
Good evening, my respected professors, seniors, and my fellow friends. Today, myself, Shruti Manna, and my group members Meghananda Nayak and Disha Biswas are present here to present our hosted presentation on the topic colonization of plastic pellets by marine bacteria based on its polymer type. Now, as we all know, that plastic pollution is one of the major environmental problems that our Earth is facing these days. Now, plastics pose a threat to the marine life also, and also to the marine microorganisms. Plastics is now by 2050, we all are assuming, or the studies have said, that there will be more plastics in the ocean water or the water bodies than the fishes. Now, through this poster, we have showed how small plastic pellets comprised of different polymer types by marine microorganisms, specifically bacteria. Plastic pellets is basically pre-production of the plastic, which can also be used as raw materials for the production of plastics. Now, different bacteria, they have different polymer types. The main significance of this is that it can degrade and colonize the plastic. Now, the rest of the part, I would like to pass it to my group member, Disha Biswas.
তোরটাও বলেছিল ও হালকা একটু আর্টিফিশিয়াল ইন্টেলিজেন্স আর এই তো আস্তে আস্তেই বলছে মোটামুটি বলেছে
Thank you, Shruti.
আমি
Now,
সত্যিকার আওয়াজ পাইনি
the impact that plastic pellet had on varying levels of colonization and degradation by marine bacteria. For example, we have observed that polyethylene pellets were more resistant to bacterial colonization and degradation than polystyrene pellets. This difference in colonization and degradation rates could be due to the very chemical properties of these plastics, making some more attractive to bacteria than others. Also, our findings have important implication for the development of effective strategies to combat plastic pollution in our ocean. By understanding which type of plastics are most susceptible to bacterial colonization and degradation, we can develop strategies or solutions to break down these plastics and reduce their impact in the marine environment. The study further highlights the potential of marine bacteria to play a role in bioremediation of plastic pollution. Here we can see that the study provides important insight into the complex relationship between plastic polymer type, bacterial colonization, biodegradation, which addresses the global environment concern of plastic pollution. Now, Meghananda Gayan will continue further.
না না ওটা দেখছি না আমরা তখন দেখছিলাম
আচ্ছা এই দুলিশ না বাবা
দেখছিলাম চিন্তা করছিল
দেখবি তারপরে এটাই অ্যাডভান্টেজ
The process consists of
সকালবেলা লাইভ হয়েছিল
attachment, bioregulation, and domination. The first stage is actually the bacteria in the water that's attached to the surface of the
plastic pellets, and the next, once they're attached, the bacteria produces some extracellular polymerase substances, which actually forms a matrix around the bacterial cell and thus form the biofilm. The biofilm protects the bacteria from any sort of foreign conditions. And next, the bacterial community of the biofilm produces some enzymes. The bacterial community of the biofilm produces some enzymes which break down the chemical bonds of the plastic pellets and thus it leads to the degradation of the plastic pellets. It is to notify here that the colonization and the degradation obviously depends upon the different types of the polymer. For example, polyethylene is more resistant to degradation and colonization, whereas polystyrene is more readily degraded. Next is that I will discuss about the potential future. The potential future has many aspects in this here because first one, further studies should be conducted about the factors which influences the colonization and also to stop the plastic pollution. Public awareness programs and education programs should be held. Now I would like to hand it over to Sri.
Thank you. Now for the conclusion, we would like to say that our study, it highlights the importance of considering polymer type or the identification when examining the impact of the plastic pollution. Now, by continuing to explore the potential of marine bacteria to break down the plastic pellets, we are one step closer of eradicating the plastic pollution. Thank you.
Okay. So what is your suggestion? I mean, how to solve the problem? There is a problem about the diabetic element of the plastic waste. So ultimately, should we ban plastic or there should be appropriate management of the plastic?
Thank you for the presentation. Now I would like to introduce to you the last presenters for today's event, Manisha Sharkar and Adrika Sharkar, who will be presenting on the topic Cryonics, Technological Fictionalization of Death.
Respected dignitaries, respected teachers, seniors, and all our batchmates.
না নেই ভাই কত সুন্দরভাবে শুরু করে আটকে দিল আমারই টপিক আমি বলছি আগে বিয়াহি দাদা এটার পর শেষ কতবার বলেছি আমি ম্যানেজমেন্টে আমাকে বারবার কেন প্রশ্ন করছিস এরকম আমি শুধু টেকনিক্যাল
A very hearty good evening to one and all present over here, respected dignitaries, teachers, seniors, and all our batchmates. Today, I, Adrika Sarkar and Anisa Sarkar, are representing our poster based on the topic Cryonics, a Technological Fictionalization of Death. Handing over the mic to my partner to start with the introduction.
How about the idea of a dead body getting back to life? Well, cryonics can help. Cryonics is a method of freezing and stooling legally dead individuals at a low temperature with the hope that resurrection may be possible in the near future. This process aims to store neural information and potentially revive them at a time using medical technologies. The preservation is done at a temperature of -196 degrees Celsius in liquid nitrogen. However, only few biological materials can be frozen at this temperature without affecting the cell viability. This picture here shows the mechanism of cryopreservation. Now, I pass on the mic to Africa to explain the significance.
One of the most significant area of cryonics is its aim to preserve a dead body so that the root cause behind the death can be cured with technology. Other facts are like the research and development behind cryonics influence society in a very positive way. For example, cord banking. Let us take the example that a baby is born and the placenta of the baby is stored with the help of cryopreservation so that later, if the baby develops with blood cancer, the stem cells present in the placenta, healthy stem cells that can be used for replacement of the cell. Coming to the mechanism of the flowchart, let us take the example that a person is dead and is also legally declared dead. Then the mechanical, then with the help of mechanical characteristics, respiration and blood circulation is carried on. Then the person is stored in liquid nitrogen at minus 196 degrees Celsius and stored with cooling. Blood and sufficient amount of water is removed from the person's body so that it can be stored. Next, handing over the mic to Anisha for the further future exploration.
Cryonics has numerous applications in space sciences, electronics, automobiles, manufacturing industry, agriculture, etc. Cryogenic detectors help determine the requirements in terms of operating temperature, stability, and architecture of the payload system. Currently, there are 3 cryonics in US and 1 in Russia. Now, the challenges will be explained by Adrika.
Even if cryonics is one of the most interesting field of research, it has still a lot of challenges. The most highlighted challenge can be the tremendous amount of cost involved in this procedure. Other challenges can be the ethical issues, cryodamage and revival. Revival involves many medical, technological requirements before it becomes viable.
Moving on with the future explorations, we have cryonics explored in future in fields of medicine, space travel, deep sea fishing, etc. Also, recently the old tires were frozen in liquid nitrogen and formed to separate steel and rubber for recycling.
Coming to the conclusion, cryonic represents an unproven medical technology that aims at saving lives. Cryonic holds the view that if current medical technology fails to keep one alive, one can choose to be preserved in liquid nitrogen in the hope to restore one back to life. Thank you for your kind patience.
Any question from audience? So one question from my side is at what temperature you are telling liquid nitrogen, but that nitrogen will be liquefied at what temperature? So for preservation actually in liquid nitrogen, the other environment is presence of oxygen, the purity of nitrogen in the body. And depending on the different coolant actually, how long you can preserve, that is also important. Nitrogen is universally used. Any other coolant may be possible for the preserving of okay.
Thank you so much. And that was the last group for poster presentation. With this, we come to the end of our poster presentation session. I would like to extend my sincere gratitude to Dr. Anand Jana sir for judging this session. Thank you so much, sir. Next, we will be starting with a valedictory session in a while. I would request everyone to be seated and be patient, please.
এখনো কি আসি
ওই রোহিত এদিকে তাকা আর আমরা নেই
না ওর পরে ও ছিল না আমি তো সেটাই বুঝতে পারিনি কি বললি वो नहीं बोला सॉरी
এই আজকে কে বলতা তোদের যে লাইভ বলে একবার পরে রেকর্ডিংটা দেখিয়েছি এরা হয়েছে
অনিশার বেলা ছিল
অনিশা এখানে বলে যাচ্ছে পেছনে পোস্টার
ভাই সৌরদীপ তোকে নিয়ে এতটাও বদন না সৌরদীপ জীবনে অনেক সমস্যা আছে তোকে নিয়ে এত বদন না মানে এমন করছে সৌরদীপে যেন সারাক্ষণ ওকে নিয়ে ভাবে
আচ্ছা ঠিক আছে করলাম লাইক করতে এবার পুরো দিন এমন বলছি লাইক করে পুরো
এখনো কি হবে
ঠিক আছে এবার এটাকে ক্যাশ করতে বল চুপ করতে গল্প
কি হবে
Now I would request the organizing committee members to come up on stage one by one. First, I would like to invite Dr. Shubhrama Gupta ma'am, Head of the Department of Biotechnology, our chairperson for NHBD 2023.
ভাই আমি আর পাচ্ছি না ঠিক আছে আমরা ফেস করতে বুঝতে পারি তোরা বাকি একটা দেখি আমি দেখি এইটা লাইভ করবো সুপার না লাইভ করবো তুইও এরকম রাধা ওই লাইভ হচ্ছে ও অফ অফ করবো এই 360 ঘোরাবো না না এইটা আরে না তুলে নিয়ে দেখি চলো একবার ব্যাক স্টেজে জানা আছে সবাইকে দেখি এই হয়ে গেছে আমি ভালো মেয়েটা
কি বললে এটা